Good afternoon, Dr. Burman.
Good afternoon, sirs.
Good afternoon.
Good afternoon, sirs. Good afternoon, sir. Good afternoon.
Sir. कहाँ पर? ठीक है.
मैडम नमस्कार.
यस सर, नमस्ते. Our jury members have joined, Dr. Jha.
Yes, ma'am. I have joined. VC Roy.
Welcome, sir. Welcome back, sir.
Thank you. Thank you.
Madam join.
Welcome, sir. Welcome, Dr.
Thank you,
Janki
ma'am.
Ram.
Good afternoon.
Good afternoon.
Rao garu bagunnara?
Sir, good afternoon.
Good afternoon, Jasab. How are you?
Fine, sir.
Nice to see Burman.
कैसे हैं सर आप?
Fine. चल रहा है. मंजे ಅವರು ಚೆನ್ನಾಗಿದ್ದೀರಾ?
Yes, sir. Yes, sir. Yes, sir. Yes, sir. Nice to see you, sir.
Nice.
Dr. Rao साहब ज्वाइन किए हैं.
हाँ, राव सर.
माथुर साहब.
Manjay sir, good afternoon.
Sir, good afternoon,
Rao
sir.
sir.
राव सर, माथुर साहब.
अभी
माथुर साहब तो अभी दिखेंगे.
यहाँ पे गर्मी है, आपको उधर ठंड है.
Yes, sir. Yes, sir.
Correct है राहुल गाరు ఇక్కడ ఏమో వేడి. Correct है जानकरू sir.
हाँ, यहाँ पे फैन चल रहा है.
फैन चलता है, रात में एसी चलेगा सर.
कितना डाइवर्सिटी है कंट्री में.
Yes, ma'am. Yeah.
डॉक्टर
It is
रामामूर्ति,
unity, sir.
वेलकम बैक सर.
Ramut sir, nice to see you, sir.
So you are all saving a lot of money.
लंच भी नहीं है.
Not even giving a cup of tea.
खाती है.
पूरा वर्चुअल में.
एवरीथिंग वर्चुअल.
वर्चुअल में चल रहा है.
He
Sir,
also
before-
virtual sir.
Yes, sir. Before, sir, we begin, I would like to just request all the jury members. So kindly finalize for MSc students scorecard today itself and send us, sir, either through mail or through WhatsApp, what is convenient, whatever is convenient, sir.
That will be
So
done.
we will do from our side, our part, and keep ready, and then we will apprise you. And final results will be declared tomorrow afternoon only, sir, after all are over, sir.
So today only MSc you are taking.
MSc, yes.
Okay.
So today's result we will finalize after receiving from you the scores.
In fact, I have,
Ji,
for
sir.
those which are already finished, I have put it down in the
Good,
table
sir.
also
That's good, sir.
already. Okay.
So we will do our work here and then.
Yeah, in
You
fact,
can do,
immediately
finally
after
you
the
can do
presentation,
moderation yourself. Okay.
Yeah.
And you put the
immediately
thing-
after presentation, I'll send it.
Yes, sir. Thank you.
Kanikram garu,
Sir. Yes, sir.
bagunnara, etla undi sir?
Fine, sir. Going on fine. Randi.
Yeah, under your guidance, sir, institute చాలా prosper అవుతుంది. నమ్మకం మాకు.
Trying our best.
Definitely, sir. మీకు ఏమైనా ఇది కావాలంటే we are always ఉన్నాం సార్.
Definitely, sir. Yeah.
తప్పకుండా.
Call you. Yeah.
Yes, sir. Thank you.
Thank you, Ramurthy, sir.
Yes, I have seen you.
Wow.
Have you colored your hair to be white or it has become white?
No, value. I stopped value addition.
Oh.
Natural farming.
Okay.
Natural farming, sir.
Okay. No, I think once you become vice chancellor, these things come.
Oh, no, sir.
White, silver, then gold.
All spoon colors, sir.
Sir, Chalapati paper got published. Thank you.
Yes, correct. Okay.
He's a good scientist.
Yes. Okay.
Excellent. He is doing that excellent job.
आप ज्वाइन नहीं कर पाए? उसी में ही है, उसी में ही नीचे 230 लिखा है ना, ऊपर 930 है ना, नीचे सेकंड लॉट है.
Good afternoon.
Good afternoon, sir.
Good afternoon, sir.
Welcome, sir. Welcome, sir.
So did you have lunch or you have come back quickly?
There was no way out.
Okay.
Good afternoon, sir.
Good afternoon, Dr.
डॉक्टर
Should
बर्मन,
we begin, sir? Dr. Burman, are you ready?
माथुर साहब को लिंक नहीं मिला, एक बार थोड़ा सा डिफिकल्टी
देखिए।
उनका।
Yes, sir, I am ready. Yes, madam.
नहीं,
माथुर साहब को लिंक नहीं मिला है, डॉक्टर बर्मन.
मैं एक बार दोबारा भेज देता हूँ.
दोबारा भेज दीजिए.
हाँ, अभी भेज.
So we are on time?
Yes, sir. Sir, one of the jury member is yet to join, sir. We're
Yeah,
waiting
no,
for
I
him,
mean,
sir.
so far we have been doing pretty
Yes, sir,
good, and
pretty
I
good.
think we should be able to finish it.
Ji.
And it was a good idea to hold it for 2 days. Otherwise, it would have been very hectic.
Yeah, I'm surprised, Ashok, that you could finish earlier in one day.
Yeah, but it used to go very late, sir. Sometimes 9, 10, 11 o'clock, up to 11, we will finalize the results. So it used to be, yeah, sometimes it has gone that late, yes.
Yeah, well.
You can adjust your camera a bit, sir, because you are seen very little. Yeah, now this is perfectly fine. That's good.
सर नमस्कार.
Namaste, sir. So,
Dr. Mathur is about to join, sir. Yeah. Dr. Burman?
मैंने लिंक भेज दिया है सर.
भेज दिया.
What? My bad. Dr. Jha, you can take the attendance of the 16 presenters.
Nine.
Dr. Jha?
Now 9 presenters, sir.
No.
Well,
Okay. Remaining
ones. Yes.
Microbiology, Aditi.
Present, sir.
Molecular biology and biotechnology, Priyanka Kumari.
Yes, sir.
Plant pathology, Akshay Kumar.
Yes, sir.
Physiology, Pratik HP. Pratik. Post-harvest technology, Soumya Sri.
Present, sir.
Seed science and technology, Sahil. Soil science, Suri Prakash Yadav. Vegetable science, Rakshita. Water science, Ankit.
Present, sir.
Professors of seed science and technology and vegetable science, kindly ensure that your students will be joining soon. So far, they have not joined.
He is here.
Okay.
He's already joined.
Okay. From vegetable science, Ms. Rakshita.
Yes, sir.
She has joined.
Dr. Behra, you are muted. Please tell whether your student is there or not.
No, sir. She is not joined.
She is not there. I'll just call her.
All right, Dr. Jha, let us start with
Dr. Mathur is joined.
Those who are present. Yes, Dr. Mathur is very much there. Those who are present in that sequence, you take first, and those who have not come, they will join later. They will go in the back.
So we once again welcome post-lunch session, each one of you, and welcome to all the jury members. So we have now 9 presentations for the MSc students. So over to Dr. Jha to take up the further proceedings.
Thank you, ma'am. Good afternoon to all of you. We are going to start our second session now, and from discipline of microbiology, I invite Aditi.
On a very optimistic note by Jenny Han, I, Aditi, welcome you all today. Well, the entire story of Green Revolution brought with it immense laurels in the field of Indian agriculture. But along with it came a Pandora box of an unexpected paradox in terms of an increased gap between legumes and cereals production, The skyrocketing values of NPK ratios, the indiscriminate use of chemical fertilizers, and this entire story can be well visualized in the case of a very important element called nitrogen. Therefore, this whole background helped us to realize that yes, there is a dire need of a balance which was termed as the flamboyant integrated nutrient management. Pulses, if I talk about, fall beautifully into this realm as on one hand they help fighting the malnutrition and on the other hand they enrich the domain of soil with biologically fixed nitrogen in the form of an inherent process termed as symbiotic nitrogen fixation. Therefore, it is one crop which in two ways has multiple benefits. In the context of Indian economy, gaining knowledge about the productivity constraints of a very important oilseed crop known as soya bean becomes very much important. Accordingly, it is a very well-known fact that the presence of externally applied nitrogen in excess interferes the symbiotic nitrogen fixation, and it becomes very crucial to understand the whole nexus at a molecular level, and therefore a study on Indian genotypes is very well crucial in the present scenario. The title of my research work was Symbiotic Effectiveness of Inoculation with Bradyrhizobium species on soybean genotypes under different nitrogen levels. With my chairperson being Dr. K. Annapurna ma'am, I worked with two major objectives. First one being screening of soybean genotypes for nitrogen uptake at different levels of nitrogen under controlled condition, and the other being response of soybean bradyrhizobium symbiosis to nitrogen application on nodulation and nitrogen fixation of selected genotypes. So moving on, the first objective was the initial screening process. Initially, I worked for an hydroponic experiment with 6 genotypes and screened them out at 3 nitrate levels indicating low, medium and high nitrate level. I worked with nitrate as it is the most inhibitory in nature towards the SNF process. Similarly, on a similar ground, I worked as an N-free pot experiment with 6 strains and screened out the best responsive strain amongst all and understand the strain genotype synergy for the overall dynamics. Also, the most suited level with respect to the genotype response under hydroponics was chosen as the representative nitrate level. The hydroponics realm helped me to visualize very beautifully that how different genotypes they responded differentially to different levels of nitrogen. And on compiling, I could see that PUSA 9712 was one genotype which caught the eye as it outperformed the others in majority of the parameters. Also, 4 millimolar nitrate was chosen as the representative nitrate level to work with. Well, in the N-free pot experiment setup, the differential responses was clearly observed in terms with the strain genotype combinations, and accordingly, PUSA 9712 Cas1 combination proved to be the best amongst all. The strain Cas1 also outperformed others in different parameters as well. Therefore, as a whole, this is the tabular form depicting the overall performance of strains with respect to different parameters and the performance of genotypes with respect to the individual Cas1. So, the major take-home message from the first objective was PUSA 9712, Cas1 and 4 millimolar. Moving on, the next and final objective was response of soybean bradyrhizobium symbiosis to nitrogen application on nodulation and nitrogen fixation of selected genotype, where the 4 millimolar nitrate was provided differentially, one as basal along with the Cas1 inoculation and the other 15 days after the Cas1 was inoculated. The entire rational thought process behind this differential application was where nitrogen was applied as basal, it was expected that it would affect the entire process of nodulation as well as nitrogen fixation. But where nitrogen was applied 15 days after inoculation, it would affect the further events of SNF as nodulation had already taken place. So, moving on, I stored the samples for their general parameters analysis and RNA extraction and differential gene expression. Accordingly, samples were analyzed at 7th, 18th and 35th day after inoculation, and the best combination was seen as N-Cas1+. On compiling and comparing with all the tabular forms at the different time intervals, I could clearly infer that the most drastic effect was observed where nitrogen was added basally. The similar thing was seen in the case of 35 days after inoculation as well. N-Cas1+ was the best combination amongst all at all time intervals. This trend was observed for majority of parameters. So the next step was moving further, extracting the RNA, preparing the cDNA, and then doing the qPCR analysis of the early noduling gene E040A. This gene was involved in nodule organogenesis and hence is considered as an early noduling gene. The respective fold changes at 7th day after inoculation and 18th day after inoculation were observed as well as tabulated for further references. The most drastic decrement was observed in the condition where nitrogen was added as basal on 7th day and on 18th day where nitrogen was added 15th day after inoculation. Therefore, based on my second objective, I could observe that when nitrogen was added basally, pronounced effect was seen in the plant parameters as well as nodulation characteristics as against where nitrogen was added 15 days after inoculation. The fold change at 7th day was greater than the fold change at 18th day and the effect of nitrate was short term in nature. I could also infer that higher doses at the time of nodule initiation had more inhibitory effect than that of the case where nitrogen was added after nodule initiation has taken place. This gives us a concept of developmental window that when the plant is trying to establish itself, if an excessive of nitrogen is added, it would hamper more profoundly the biological nitrogen fixation process. So, what all future inferences could be taken from this experiment? An insight into the expression analysis of an important marker gene was done. There is a scope of a detailed work on similar grounds on other marker genes as well. This differential gene expression analysis at different time intervals can help us to visualize that what can be the aftermath of nitrogen application on the expression dynamics at a temporal scale. And this detailed work can help us to understand further the effect of genotype on the synergy of BNF and the application of mineral nitrogen. And this would help us to visualize the genetic regulation of nodule formation with respect to external nitrogen availability. In this era of genome, proteome and transcriptomics, a detailed transcriptome and proteome analysis at different plant stages with respect to variable factor of genotype as well as strain can also help us to envisage that at molecular level, what is the dynamics which deals with the SNF process. Also, this study can help us to gain a new insight into the recommendation of fertilizer doses. Why? Because the levels at which both the phenomena occurs in a synergistic manner would be more preferred for increasing productivity in soybean. Higher doses at the time of nodule initiation has more inhibitory effect than that of the case where nitrogen is added midway of the plant nodulation process. My life at IRI was a perfect amalgamation of research as well as extracurricular activities as I participated in various activities. I also have a personal blog termed Expressions where I write poems, and as of now I am pursuing PhD in Biological Sciences from CSIR CCMB Hyderabad. Summing up, I express a very warm token of appreciation towards each and every milestone which has covered the research life here at IRI. And with this very beautiful quote by Immanuel Kant, I now reach towards my final destination. Thank you so much.
Open for discussion.
Yeah, I think I compliment Aditi for a good work and also our advisor, because it has applications, especially with regard to nitrogen fertilizer application, especially the stage and time of application of nitrogen and its dose. The study will go a long way in helping the fertilizer recommendations for soybean. Anyway, how did you measure this nitrogen fixation, Aditi? Did you take up any studies?
Yeah, I did the ARA estimation as well as measured the nodule number, nodule weights at the 3 different time intervals.
You used Estlin reduction technique.
Yeah, ARA activities.
No, what was the best time you could find for application of nitrogen so that there is a synergy between symbiotic nitrogen fixation and nitrogen application?
As if I consider the 4 millimolar nitrate, it was the base of my study. So 15 days after the inoculation has been already done, when I applied the nitrate, I could see that after 35 days also there was nodulation, nitrogen fixation, not as much as in the case where no nitrogen was added, but there was nodulation as well as the area activities was visible. So the most deterrent case was where nitrogen was added basally along with the Cas1 inoculation.
Was it pot culture or was it under incubated conditions?
Yeah, it was under greenhouse conditions.
Greenhouse conditions.
Yeah.
Okay, that means you have taken into consideration the soil interactions with rhizobium also. Soil plant microbe interactions.
Yeah.
All put together, you could drive that.
Yeah, all put together in a whole dynamic scale. Yeah, it was a short basic experiment, but in long run, if we can go further with different marker genes, we can get a clear picture that what actually is happening inside the realm of when excessive nitrogen is being added.
Thank you.
Thank you, sir.
Aditi, if the chairman permits, may I ask you a question? Chairman, sir, can I?
Please go ahead, sir.
I have 2 questions, Aditi. One is, did you find any correlation between the nodulation and the ARA activity? One. And second is, what is your recommendation, say for soybean? How many splits we should go for?
Okay, so the answer for the first question, yes, I did saw correlation between the nodule growth and the area activity, but it was also depending upon the species wise, the genotype wise which I chose. For example, in one genotype, the nodules were more in number, smaller in size, and the area activity was less. But in another case, nodules were larger in size, smaller in number, with a greater amount of area activity. So what I could infer was the nodule weight, the whole nodule weight was affecting positively for the area activity. And as for the second answer, if we do a detailed split up, like I did a splitting only at 15th day. If we do a detailed, like for example, we are doing it at 15th, or another we can split it at 35th day when the plant is about to flower. Then if we can do a detailed analysis, then I think so a picture can be very clearly seen. And as of now, I would surely recommend that don't give the excessive dose of nitrogen at the very starting, at the very initial point when the plant is trying to establish itself in the soil dynamics.
Because this is true not only for say legume crops, but also for other crops, the starter dose, because till the time around 7 to 8 days or 9 days when the plant is still establishing, the plant will not be using whatever the basal dose we give. So, and for legumes, we don't apply too much of nitrogen at all, first of all. So
yes,
this is very, very tricky issue. And if it is dependent on the genotype, then recommendation will be little hard to come by.
Exactly, yes, sir. It will depend on the genotype wise also, because I could very clearly see that how different genotypes respond differentially to even the nitrate application and to different timings as well.
Okay, thank you.
Good presentation, Aditi.
Thank
Good
you, sir.
presentation with a lot of clarity. One question I have.
Yes, sir.
You mentioned that use of higher dose of nitrogen when you use
Yes,
it at the nodule initiation stage.
Yes, sir.
Having more inhibitory effect.
Yes, sir.
What's the implication of this finding?
The implication goes like this. For example, I am a farmer and I'm completely relying on the external doses of nitrogen. And if I'm giving an excessive dose, I'm completely trying to shut off the biologically nitrogen fixation process which is going at the host level. And as per different studies, it has been visualized that both SNF as well as the external nitrogen is applied in harmony to give the overall shoot up in the productivity. So if I am able to understand that, okay, if at one point scale, both the processes are occurring in harmony, I can somehow assure that, yes, my productivity can shoot up and the constraints can be taken care of.
Okay, has it been published? This finding has been...
No, it has not been. No, because the thing is that we wanted to see its reproducibility per se, and its reproducibility can be checked if we do it in multiples. So the next experiment is in line. Maybe in a month or so, we'll get the final detailings, and then we'll go for the publications per se.
Okay, good work. Nice.
Thank you, sir.
Aditi, one clarification I seek, it is on what you said and what Dr. Ramamurthy also mentioned, that, you said an application should not be given at nodule initiation stage.
Yes.
So my question to you is, can you identify the nodule initiation stage in legume crops, or it is a continuous process which happens throughout the crop period, and the old nodules, they will die and the new ones will keep coming? So if you believe that it is a one-time trigger or it is a multiple time trigger of nodule initiation?
So that depends. There are different species in which it is a continuous process, and there are some species in which it is a discontinuous process. Once in a lifetime, the nodules begin and then they senescent and die. But in some other, it goes in a synchronous way. Some species, and in that way, some nodules they senescent, and the other nodules they again take the process further. So it depends on the species with which I'm working.
Thank you.
Thank you.
What is the situation in soybean?
Soybean is non-synchronous. It happens once initially, and then the nodules they die.
It's synchronous or non-synchronous?
It is non-synchronous.
It's not at a time. It's at different times. Non-synchronous would mean not at a time.
Oh, sorry, I'm speaking it in a different way. It is synchronous. It occurs once at a time, and in other species it occurs differentially.
No, the root, you talk about 15 days application having no effect. Now in 15 days, the whole root system will not have grown.
Yeah.
So that means roots that come at the time, they will not have any nodules.
No, the thing is that the taproot has already been formed and the nodule initiation points have already been there. So now as the root will expand its domain, the nodule initiation points, the nodules would form and then they will expand as the root itself expands.
Oh, God. Okay. Any more questions? That was nice presentation, Aditi, and thank you very much.
Thank you so much, sir.
Thank you, sir. We move on now, and I invite now Priyanka Kumari from Molecular Biology and Biotechnology.
Good afternoon, all and one present here. I am Priyanka Kumari presenting for MBP division. According to UN report 2019, India is going to be most populous country with 1.6 billion population in 2050. By 2030 itself, we will surpass China, and to fit this huge population, we will need 333 million ton food grains. Among food grain, wheat contribute a major portion in our diet, and we will need 140 million ton by 2050 with present level. This is true that with Green Revolution, we got huge production in wheat and rice due to development of high-yielding varieties which require intensive tillage practices, higher irrigation, high fertilizer and pesticide application. Due to all these practices, favorable microclimatic condition was created. As a result, one of the most omnivorous wheat, Phallus minor, became dominant. This wheat compete with wheat for nutrient and space, and with time it became resistant to all kind of herbicides. Among all nutrients, nitrogen is very important for wheat. Wheat take nitrogen in the form of nitrate, but if phallus minor grows into field, then it competes with wheat for nitrogen uptake and some nitrate get lost to environment. The loss of nitrate to environment can be in the form of leaching, denitrification, and immobilization. And the nitrogen use efficiency of wheat is only 30 to 40%. And if phallus minor is there, then there is a loss of 28 kg of nitrogen and potassium and 3 kg of phosphorus per hectare. So we have to work on wheat architecture so that it can resist the phallus minor infestation, as applying huge amount of fertilizer and herbicide is not a proper solution. So the nitrate which are taken up by wheat moves inside through transporter like NRT1 and NRT2. NRT1 is called low affinity transporter and NRT2 is called high affinity transporter. Under NRT1, NRT1.1 is reported as a dual affinity transporter and Arabidopsis NRT1.1 is reported as a transceptor, that is both transporter and receptor activity, and it influences lateral root development. So research gap identified was root system architecture study of wheat with phallus minor is poorly understood in unlimited condition, and it will be interesting to study ATNRT 1.1 ortholog in wheat which influences root architecture. So my title of the thesis was Understanding Self and Non-Self Root-Root Interaction on Root System Architecture and Expression of Putative Nitrate Transceptor Gene in Red Wheat under Nitrogen Limited Condition. To which my objectives were to study modulation of root system architecture and uptake in wheat in presence of growing phallus minor reg and effect of non-self root-root interaction on expression of AT and RT 1.1 ortholog in wheat. Under objective 1, individual phallus and wheat seeds were grown and seedling were raised applying MS medium with CaNO3 at 4 millimole as the external nitrate source. When they achieved a comparable biomass, seedling were brought together in a pot in two combinations. One contained phallus and wheat seedling and other containing wheat and wheat seedling. This setup was for N optimum condition. For N limited condition, 0.04 millimole CaNO3 was applied, and this setup was done in two sets, one for root system architecture study and other for expression studies. For RSA and biomass study, plants were uprooted from pot, washed in tap water and separated into shoot and root. Press weight of shoot and root were then taken. After the same root is used for scanning in Windows software. Then length of shoot and root were measured. After that, shoot and root were packed separately and kept into oven at 42 degrees Celsius. After 10 days, dry weights were measured. After root scanning, roots were analyzed, and these pictures shows the scan root images of wheat and phallus minor before interaction, and it can be seen that they are comparable in root system architecture. This picture shows the interaction of wheat with phallus and wheat itself in an optimum condition, and it can be seen that there is not much difference. These two pictures shows the interaction of wheat with phallus and wheat in unlimited condition, and the difference among roots can be seen. After this, 5 root traits were studied. First one was total root size, and from this graph it is seen that TRS is getting increased in presence of phallus minor in an optimum condition also and in unlimited condition also. Next trait was main root path length, which is also getting increased in presence of phallus minor in unlimited condition. Third trait studies was lateral root density, which is also getting increased in phallus minor presence in n limited and n optimum condition. And the next trait studied was first order lateral root number, which is also getting usually in presence of phallus minor in unlimited condition and to some extent in an optimum condition. The last trait was second order lateral root number, which has a similar increment pattern as first order lateral root number, that is more increment in presence of phallus minor in unlimited condition as compared to the n optimum condition. After root scanning, biomass has been measured, and it was found that shoot length was highest for wheat-wheat interaction in an optimum condition, and it was lowest for wheat-phallus interaction in unlimited condition. Next was root length, which was found highest for wheat-phallus interaction in unlimited condition. Then shoot phase was taken into consideration, and it was found wheat-wheat interaction in an optimum condition has the highest shoot phase as compared to wheat-phallus interaction in an limited condition. Next, root phase was taken into consideration, and it was found that wheat-phallus interaction has the highest root phase as compared to the wheat-wheat interaction in an optimum condition. Next biomass measurement was for shoot dry weight, which was found lowest for wheat-phallus interaction in unlimited condition, and the root dry weight, which was found highest for wheat-phallus interaction in an limited condition. Under objective 1, nitrogen content was also measured. For this, dry root and shoot were taken and lyzed homogeneously. Then they were mixed into one Falcon tube, and total nitrogen was measured by Zeldar method. Some sample was also sent for isotope ratio mass spectrometer to check delta N15 content. Total nitrogen measured through a Zeldar method shows that it was lowest in N limited condition in presence of phallus minor, and it was highest for wheat-wheat interaction in an optimum condition. This shows that the phallus minor is competing with wheat for nitrogen uptake, and this effect is more pronounced in an limited condition. Next, delta N15 value shows that the wheat phallus interaction in unlimited condition has the lowest value, that is 0.335, which again proves that phallus minor has more pronounced effect on nitrogen uptake activity in unlimited condition on wheat. Under Objective 2, first AT&RT 1.1 ortholog has been searched in wheat genome. This search results in 13 orthologs in wheat. Also in case of rice, 3 NRT 1.1 has been reported, but only NRT 1.1b acts as a nitrate transceptor. So 13 orthologs of wheat were blasted with rice NRT 1.1b to find closest nitrate transceptor, and a phylogenetic tree was constructed, which saw 4 NRT 1.1 in all 3 genome of wheat, which were grouped as a TANPF 6.4. Next, RNA was isolated separately from seminal and lateral roots of wheat, and then cDNA was prepared and RT-PCR was carried out with homolog specific primers. After RT-PCR analysis, it has been found that TNPF 6.4a has the highest expression in seminal roots and lateral roots. In seminal root, it expresses more in case of an optimum condition, but in lateral roots, it expresses more in case of an limited condition. Also, the TNPF 6.4b1 has the negligible expression among all 4 homologs. For the differential expression observed in homologs, we clone 2 kb upstream sequence of 3 homologs. For this, DNA is isolated from canine 107 leaves and PCR amplified with homolog specific primers. After PCR amplification, 2 kb bands were eluted and ligated in PZ1.2 plantain vector. Then this is transformed into BS5 E. coli cell. Transformed pair selected based on the ampicillin resistance. From positive transformed, plasmid DNA was extracted and restriction digestion was carried out using BGL2 enzyme. This is a 3 kb vector band and this is a 2 kb insert band. After that, clone were sent for sequencing. After sequencing, sequences were submitted to gene bank and cis regulatory elements were analyzed using nuclease and ProCyte database, which gave an image showing respective position of CREs. After analysis, it has been found that CREs not only varies in number but also in presence and absence in the homologs. The conclusion that has been drawn from my studies are that phallus minor predominantly modulate the RSA and translocate shoot biomass to root under N-stop condition. Also, nitrate uptake and N15 influx drastically reduces in presence of phallus minor, that is non-self root. And TNPF 6.4 is the most sensitive among all 4 homologs, which expresses more in seminal root in N-optimum condition and in N-limited condition expresses more in lateral roots. Also, the cis regulatory element analysis shows variation in terms of number and also in presence and absence, which might be the cause of differential expression of the 4 homologs. So future prospects from my study could be differentially expressed putative nitrate transceptor will further be validated for its nitrate sensing function, and upstream sequence could also be characterized for its root specific role. My few achievements are: I have been awarded National Talent Scholarship, Junior Research Fellowship, University Gold Medal 2018, and I have cleared DVT-CRF, CSIR net and ICAR net. So I like to acknowledge my chairperson, advisory committee members, Professor MBB, Project Director NIPV, PG School IARI, ICAR, Junior Research Fellowship, parent, friends, seniors and juniors. Finally, I would like to end my presentation with a quote of Dallas Euler that if we can get the biotechnological traits, we may be able to turn wheat around. Thank you.
So, can I ask a question?
Please.
So it was a very good presentation. I have 2 questions. One is what is the reason for selecting a 42 degrees Celsius for drying the root? And another one question is how the NRT 1.1 play a role in root development?
For the first question, there is no specification for 42 degrees Celsius. Means it's means I have selected based on what is used in my lab. That is 42 degrees Celsius. It is set. So for it won't get more dried or less dried. So we kept at 42 degrees Celsius, and then after 10 days the dry roots were taken. So there is no specification that we have to do on 42 degrees Celsius itself. We can increase or 5 degrees Celsius less or more, that's okay. And for the second question, that is NRT 1.1 means role in root development. So NRT 1.1 in case of Arabidopsis, it has been found that it has a role in some molecular mechanism, that is by oxygen signaling, that if the nitrate is present in more condition, then it will help in the oxygen signaling, so that it will develop the lateral branches towards the nitrate rich patches. So it will if nitrate rich patches are there, then it will function and it will enhance lateral branches to take up the more nitrate from the soil.
Is it transport other than nitrate any other molecule?
It has been reported by some authors that it can transport glucosinolates and other materials also. It's a kind of master regulator gene. It has many functions, among which it influences lateral root development also.
It can also transport IAA, that is the auxin. That's why it regulates root growth.
Yes, sir.
Drying, you normally use a higher temperature so that the metabolism is immediately stopped. So normally we use drying about 70 to 80 degrees Celsius, not at lower temperature. Very good.
Okay, sir. Thank you.
Any question?
Sir, if you kindly permit, I have
Please.
one question.
Yeah, please.
Betty, you did N15 studies, and my question is that whether the N source that you used, was it enriched with the N15, or what was, what was it study performed with the normal nitrogen that you get it?
For N15, we use for the precise measurement of the nitrogen content which is present in the root, so means root of wheat in presence of Valdis minor. So N15 will tell us what is the precise amount which is present. So that's why we go for N15. We measured first total nitrogen content by cellular method, that's okay. But after measuring that, we send some sample for N15 so that we can know that it means precise measurement, that this much amount of N is present, N15 is present, that influx or how much getting influxed in the root system in presence of Valdis minor. So for influx purpose, that this much nitrogen is getting influx, for precise measurement, we use N15.
Priyanka, my question was very simple because normally N15 it's also a tracer stable isotope, and we use it for differentiating between the nitrogen which is derived from the fertilizer or from the soil.
Yes, sir.
Because nitrogen is there in the soil and you apply it as a fertilizer also.
Yes, sir.
Normally what we do is we use the enriched form. Say if you want to identify nitrogen derived from fertilizer, enriched fertilizer source, N15 enriched source. In absence of that, it will only tell you what is the total N uptake, but that is, you will be anyway getting it from the N total N data.
Total N uptake is
N15 will not reflect the efflux or influx. It is only uptake, and it can differentiate if your applied source of nitrogen is differentially enriched, or if you are having two sources which have differential N15 attributes, then it will be, you can, based on a higher N15 recorded, you can know what is the, from where it is coming. Otherwise, it is not a reflection on, the total N uptake or total N efficiency.
For efflux, it can give means how much getting N15 is present in a tissue, that can be checked by that N15 value. That efflux is this much is getting influx in the plant root system.
You can check, but it is not like that. Huh?
Okay, sir. Okay, sir.
Priyanka, rather it's not a question, it is an observation from me. I think you don't have any publication out of your work. Rather,
yes, sir. Because of that COVID-19, I'm left with one cloning. That's why I thought of publishing after that cloning. So I'm left with one cloning.
You have spent a lot of time in root architecture and biomass.
Yes, sir.
And then further taking them beyond NRT1 and NRT2. Rather, if you had spent more time on NRT1 and NRT2, you could have finalized it towards the end, some logical ends, and it could have brought out some publication. An observation, that's all, just a comment.
Yes, sir. Yes. Okay, sir.
Yeah, Professor Jha, we could thank the candidate and move on.
Thank you, Priyanka.
Thank you, sir.
Thank you, sir. And we move on to the presentation. And I invite Mr. Akshay Kumar HM from the discipline of plant pathology.
Good morning, everyone.
I extend warm welcome to you all for my merit medal contest presentation. First, let me introduce myself. My name is Akshay Kumar HM and I am from the Division of Plant Pathology. We all know that wheat is the most important and a strategic cereal crop for majority of the world's population. Nearly 2 billion people around the world consider wheat as a staple food. Wheat provides nearly 55% of the carbohydrates and 22% of the food calories that is consumed globally. But this wheat is affected by many diseases which are the major cause of yield loss, which in turn threatens the global food security. Majority of these diseases are caused by fungi. Among these, the Fusarium AIDS scab caused by Fusarium graminearum is the most damaging disease among all. Why is this disease considered so damaging? The reason being the cosmopolitan nature of wheat production, and this disease is considered to be at the top of the major diseases affecting the wheat with more than $7.7 billion loss annually. The fungus also produces mycotoxin that poses a significant threat to the domestic animals and humans. The pathogen also gained importance in a climate change perspective, which led pathogen to become more prevalent in the tropical countries like India also. According to the United States Wheat and Barley Scab Initiative, nearly $6 million of fund is spent on the research aspects of this disease annually, out of which 25 to 30% will be for the pathogen genetics and the variability studies, and the other aspect being the management. But in the Indian scenario, there is a lack of knowledge on variability in the aggressiveness of a pathogen and also on the host resistance in the Indian cultivars. Pathogenic and the genetic variation among the Fusarium isolates causing Fusarium head blight, and evaluation of wheat genotypes under artificially inoculated condition for resistance against Fusarium head blight pathogen. Under the first objective, collected around 110 infected wheat ear heads from the Laol Spiti Valley and Wellington, and also from IRA New Delhi. Out of these, 29 Fusarium gramineum pure cultures were obtained. All these isolates were confirmed by Indian type culture collection as Fusarium gramineum isolates. These 29 isolates were characterized for morphological characters such as color, growth rate, pigmentation, and microscopic studies for analyzing the variation in the macroconidia length and width was carried out for all the isolates. The color of the mycelia varied from light pink to the brownish pink, and macroconidia was produced in all the isolates. The spore length varied between 44.2 to 45.9 micron, and spore width varied between 5.7 to 9.4 micron. The growth rate was much higher in the Wellington isolates compared to other region isolates, and all the isolates produced pink pigmentation on the PDA media. Later, highly susceptible wheat variety HT2851 was used for pathogenicity test of these 29 isolates. Spore suspension was inoculated into a wheat plants at a mid-anthesis stage by cotton web inoculation. All these isolates were found to be pathogenic. For analyzing the pathogenic variability among these isolates, the 29 isolates were inoculated on a set of 10 wheat varieties, which would give the differential disease response, and scoring of disease data was done. For scoring of disease data, modified Japanese scale, that is 0 to 5 scale, was used. Later, mean data of percent spikelet infection was recorded at different intervals for all the isolates. After 7 days of inoculation, the isolate FGW18 resulted in a maximum disease of 10.77%, while the minimum percent spikelet infection of 5.7% was recorded with the isolate FGW1. Similarly, after 14 days of inoculation, isolate FGW10 resulted in a maximum mean disease of 57%, and minimum of 25% spikelet infection was observed with the isolate FGW7. These data reveals that there exists a difference in the aggressiveness among the Fusarium gramineum isolates on a set of wheat varieties. Later, for analyzing the genetic variability among these Fusarium gramineum isolates, we identified the SSR regions in the genome of Fusarium graminearum. For this, we took Fusarium gramineum PH1 whole genome sequence and a MESA tool. A total of 1,577 SSR markers were identified, including all 4 chromosomes, with the highest of 57% of XR repeats, followed by tri repeats constituting 17%, and the least of tetra constituting only 5%. Of these, 23 markers of 3 repeat motifs and above were selected based on the repeat motif length, it representing all 4 chromosomes. Using Primer 3 tool, we designed the primer for these 23 markers. Later, for analyzing the polymorphism of these 23 SSR markers, the DNA of all the isolates were isolated using CTAB method, and it was used for diversity analysis studies. All fusarium brominium isolates were amplified for ITS region. The PCR amplified product showed band size of 550 base pair in all the isolates. After sequencing, the obtained sequences were compared with the NCBI GenBank and found similar with the fusarium brominium ITS region in the database already available. Later, these selected 23 SSR markers were validated in the whole genome of all fusarium brominium isolates. Out of 23 markers used in the study, only 21 markers could give amplification, including all isolates. Then, scoring of alleles was done. Polymorphism information content or gene diversity values were calculated for 21 markers used in the study. PSA value varied from 0 to 0.97 for these 21 markers. And these markers could amplify 34 alleles including all isolates in total. And the SSR21 amplified 4 different types of alleles of different base pairs. For cluster analysis, Jaccard scoring of genetic similarity in the fusarium graminearum isolates was used to create dendrogram. UPGMA clustering method was used in a NTSys software. This separated all the isolates into 2 major groups. The isolate FGW27 and FGL2 grouped in the cluster A, and all other isolates were grouped in the cluster B, which was further divided into B1 and B2. The B2 contained single isolate, that is FGL1. This result indicated the existence of variation not only among the isolates of different region, but also there exists variability among the isolates collected from the within region. Under the objective 2, we screened 164 wheat genotypes for fusarium head blight resistance under artificial conditions. Data on average percent spike, spikelet, and AUDPC and relative AUDPC values were calculated based on percent spike infection. Most of the genotypes were categorized as either susceptible or highly susceptible. Based on percent spikelet infection, we found 2 genotypes HT3377 and HI1612 as moderately resistant. Based on the AUDPC values, relative AUDPC values were calculated with respect to the highly susceptible variety Sonalica, where we could find around 6 genotypes which showed slow scabbing of head scab disease. Coming to the conclusion of this study, the morphomolecular approach was used to select the 29 fusarium gramineum isolates. We could find the significant amount of variability for the pathogenicity among the collected isolates. And in the fusarium gramineum genome, a total of 1,577 markers were identified with the maximum of XR type of repeats. And these 21 markers could amplify 34 alleles, indicating the reliability of the markers for polymorphic studies. And dendrogram indicated existence of wide range of genetic variability, and we could find 6 genotypes which were slow scabbing and 2 genotypes which were moderately resistant to the disease. For the future perspective, need for focusing on chemotyping of these isolates and search of resistance in the indigenous germplasm and distribution dynamics of a pathogen need to be studied. My academic achievements are as follows. I have published 2 research papers and I have received 1 Best Poster Award. And I have presented for 2 oral presentations and 1 poster presentation, and I have documented 8 sequences to the database, and I have received ICR JRF Fellowship of 2018, and I have secured All India Rank 3 in ICR SRF 2020. I have also participated in many other national and international conferences and webinars. I would like to acknowledge my chairman and members of my advisory committee and all from the Division of Plant Pathology and PG School IRA and ICR for PG Fellowship. At last, I would like to thank you all for patient listening. Thank you, one and all.
Thank you, Akshay. And now it is open for discussion.
Yes, sir.
Maybe I should begin. Akshay,
Yes, sir.
you used some varieties to find out if they could serve as host differentials.
Yes, sir.
What was the status of that finding from that effort?
Sir, those varieties actually we selected based on the previous reports, sir.
Mhm.
Based on previous reports, we selected the varieties which could give the reaction varying from susceptibility to means highly susceptible to highly resistant.
So could you classify the isolates into a group of races or pathotypes or something? Was it?
No, sir. Actually, we need to focus more on these differentials, sir. Actually, as it is an ectotrope, we couldn't come to the conclusion. Like most of them having the reaction of similar type of reaction they were having. Like on a susceptible variety, they were giving more infection. Like that, they were giving very less isolates could give the differential reactions.
It's genetics of resistance of the host known, or it is still to be
Yes,
worked
sir.
out?
Sir, known, sir, many QTLs has been mapped already. In Durham, around 12 QTLs have been marked, and especially many QTLs has been marked in hexaploid wheat. Especially now in nowadays for breeding purpose, they are using Sumai 3, which is having FHP1 gene, FHP1 QTL. That is the major source of resistance right now, what they are using worldwide.
Yeah, one major QTL that was that what I thought was there. Others may be there, but is
there,
that
sir,
QTL known in Indian material, or someone has tried to find out?
Sir, means not known, sir, means they have tried to create elite lines in Indian Institute of Wheat and Barley Research. They have carried out certain research, and they have tried to create some elite lines using Sumai 3 and Fontana. But none of means, and right now we are in Indian case scenario, we are having two genotypes in NBPGR. Which are resistant, but no gene has been inculcated into the cultivar, any cultivar or variety, sir.
One last question, Akshay.
Yes,
you
sir.
said that you identified a number of SSR markers for the fungus.
Yes, sir.
How did you develop
sir.
them?
Sir,
are they previously reported, or you developed them?
Sir, while selecting means we have using whole genome sequence, we have we have identified all those, sir. On our own we have identified, but for selecting we use certain criteria, like we should use try repeats, and there should be a certain amount of GC content, the length should be like this. On that criteria we selected, and a few we selected based on previous, that motif was showing reproducible amount of polymorphism, some such motif related markers also we took.
Okay.
Okay, I have one or two questions.
Yes, sir.
Really, it's a good study because it's a classical plant pathology study where you have studied the pathogenicity.
Yes,
and classified the variations in their culture, and then later you have seen the reaction to genotypes also.
Yes, sir.
One question which is occurring to my mind:
yes, sir.
Is there any possibility of synchronizing all these three things together? For example, you have found out the pathogenicity in terms of, you call it as aggressiveness, I call it as virulence,
yes,
and also there are variations in their reaction to the host genotypes. If you try to put all these things together, sometimes it may so happen that there can be a kind of synchronization may happen.
Sir. If we go for like chemotyping, means actually this pathogen is more important because it produces mycotoxins. If we go for if we select such genes which produces mycotoxins, and if we go for chemotyping of these pathogens, then we may find the similarity between the pathogenicity or the aggressiveness of a pathogen with variability in the genetic basis. But we have in future we are planning for chemotyping of these isolates, sir.
Can it be tried, at least the existing data, whatever you have generated now?
Yes, sir.
Can't you try, do a cluster?
We tried, but particular means we couldn't find like that, sir, like similarity between genetic and pathogenic variability. So I have not mentioned that.
Okay, okay. Thank you. Good.
Yes,
Good work. Nice work.
Chairman, sir, shall we move ahead?
Oh, sorry. Please go ahead.
Thank you, sir. So thank you, Akshay.
Thank you, sir.
I now invite Mr. Pratik HP from the discipline of plant physiology.
Namaste everyone. My name is Pratik and I come from Division of Plant Physiology. A very warm welcome to you all. Everyone is talking about the climate change and food production today. Let us talk something different. That is about the universe. A universe of proteins within us, which is similar in many ways to the universe we live in. You'll be wondered to know that we have very little understanding about the universe within us, just like the space. Let us get into some basics. A functional unit of a protein is called as domain. In the protein family database, we have two kinds of proteins. One class of known function and another of unknown function, which often has a domain of unknown function. And of course, we are interested in the unknown. The number of proteins with domain of unknown functions are increasing in the databases. These are rich sources of novel biological roles yet to be discovered. Stress response is not an exclusion. So there is a lot of sequencing efforts in the last few years and data is piling up in the databases and it's time for us to annotate. That's why the domain of unknown function proteins is called as dark matter of the protein universe. How we found this domain of unknown function proteins is an interesting question. Initially, we were looking for multiple stress-responsive hypothetical proteins in rice. We found an interesting protein with DUF1645, which was reported to have a role in stress response as well as grain yield. Two reports from 2016 suggested that a protein of DUF1645 family conferred stress tolerance in rice as well as increased yield up to 46% when overexpressed. These limited reports solidified our goal to study these proteins of DUF1645 family in detail. With this background, we decided to take up our work on investigating the role of DUF1645 proteins in abiotic stress tolerance and grain yield in rice. Initially, we have set up three objectives. The first one was focused on genome-wide identification of DUF1645 genes in the rice genome and computational analysis. The second objective was to carry out expression analysis of DUF1645 genes under multiple stress conditions. And the third one was designing CRISPR-Cas9 constructs for generating mutants for DUF1645 family of genes in rice. Under the objective 1, we explored the computational aspects of the protein under study. Our first finding was that the DUF1645 family was evolved in early bryophytes, possibly during transmission from water to the land. We are surprised to find that the DUF1645 family is endemic to plants, and we also found that these family of proteins are highly diverse within cereals. Phylogenetic analysis suggested a huge diversity within DUF1645 protein family. We have used robust tools like RAXML to generate a phylogenetic tree with 1000 bootstrap values. We have used sequences from 13 plant species, that is total of 183 sequences, including 15 sequences from rice. The protein tree was divided into 9 groups, and it of course suggested that cereals are more diverse than the pulses. Our next goal was to study the gene duplication. We used MCS scan to map the genes onto the chromosome. We found that there is 2 pairs of tandem duplications and 2 pairs of collinear duplications led to the evolution of DUF1645 gene family in rice. We have conducted a multiple sequence analysis of the DUF1645 family. We have found that these DUF1645 genes have highly conserved MAP kinase interaction sites. This was the first clue about their molecular function. To confirm the presence of interaction sites, we employed MEME motif discovery tool to identify the conserved motifs in the DUF1645 family. We noticed that there are highly conserved 1433 interaction sites, phosphorylation sites, as well as MAP kinase interaction sites. Further, the co-expression analysis using string confirmed that the DUF1645 genes in rice and Arabidopsis co-express with MAP kinase proteins and stress-responsive transcription factors. Through thorough literature mining, we also found the evidence for interaction of a Arabidopsis DUF1645 family member with MAP kinase during ethylene and abscisic acid treatments, and We also found 1433 proteins interact with phosphorylated proteins in the signaling cascades, suggesting us that the DUF1645 family of proteins might need to be phosphorylated for their functioning. We were very much surprised to note that the DUF1645 family of genes lack introns, and they are in turn regulated by microRNAs. The microRNAs regulating these genes also are found to be stress responsive. For example, microRNA 319A. Further, to understand the possible role of these genes and the pathways they are involved in, we have conducted a thorough promoter scan around 2000 base pair upstream to the start site. In the rice and the Arabidopsis, we have found cis motifs related to hormone, stressors, development, and signaling. We have repeatedly come to know through various approaches that these proteins are rich in proline content and are highly phosphorylated, probably a post-transcriptional modification they needed for their function. Remote homology search and other approaches suggested that these proteins are distantly related to DNA binding proteins, and these proteins themselves could be DNA binding proteins. So, looking into the protein structure might provide a more detailed information. A perfect clue came in when we discovered that these proteins are highly disordered and they have nuclear localization signals, which is a usual property of proteins which bind to DNA and also the proteins which interact with multiple proteins. The intrinsically disordered structure of these proteins suggests that they are involved in a regulatory roles in plants. Our next objective was to understand the behavior of these genes under stress conditions as well as during the developmental stages of the plant. We noticed high levels of expression of these genes when there is an active cell division, for example during early vegetative stage or during the reproductive stages. Expression during the grain development also suggested that these genes express where there is active cell division. A QRT-PCR expression study reiterated our findings. We treated rice seedlings with osmotic, cold and salinity stresses for 24 hours. A few of DUF1645 genes homologs showed reduced expression. In contrary, other few showed elevated expression. This suggests us that these genes might be involved in a regulatory role in stress response. Since we have found higher number of hormone-responsive cis elements in the promoters of DUF1645 genes, we decided to look into expression of these genes in response to phytohormones. We have found that these genes are highly responsive to abscisic acid and jasmonic acid, the both of the hormones which are known to be stress responsive. Mutants are indispensable part of a study focusing on functional analysis of the genes. We decided to generate mutants for DUF1645 homologs in rice using CRISPR-Cas9 technology. We have designed totally 8 guide RNAs targeting DUF1645 homologs in rice. The guide RNAs were designed using CRISPR-P software, and we have taken into account of on-target score, off-target score, and RNA secondary structure during the designing of the guide RNAs. Initially, we have cloned the guide RNAs into PSGR entry vectors. Selection was done using antibiotics and confirmation using PCR at each step. Finally, sequencing was done to confirm the clones. After that, the guide RNAs from PSGR vector were mobilized into pCambia destination vector, which goes into the plants. The selection and confirmation at each step was followed. The final confirmation was by sequencing. Finally, we have successfully cloned 8 guide RNAs targeting DUF1645 genes into pCambia. Each construct was designed to have 2 guide RNAs targeting a single gene to enhance the efficiency of editing. Time is always short for research. 2 years is a very short duration and we have many things left to do. With respect to first objective, we need to prove the MAP kinase DUF1645 proteins interaction using BIFC or East hybrid systems. We also aim to correlate the expression data of these genes with the rice SNP data. Finally, the constructs are ready and we need to do transformation to get the mutants. Coming to the summary of our work. Our work can be classified into 2 aspects. First, role of DUF proteins in plants. Second, their role in DNA binding. These are the respective findings. DUF genes are endemic to plants and intronless. They are multiple stress responsive and hormone responsive. They regulate the grain length and are in turn regulated by microRNAs. They are intrinsically disordered and more propensity to bind. They interact with MAP kinases and localize in the nucleus. They co-express with stress-responsive transcription factor. Our study not only gives the first molecular insight into DUF1645 proteins, but it also serves as a model study for future studies focusing on the functional validation of hypothetical proteins. A short summary of what we think it might be happening. The DUF1645 proteins interact with MAP kinases and gets phosphorylated. In turn, they interact with 1433 proteins and then get mobilized into nucleus. Inside the nucleus, they bind to the DNA and they bring the desired changes in the expression level of the downstream stress-responsive genes. I feel fortunate to have got 2 international awards. In 2019, I was named as the American Society of Plant Biology Converon Scholar Award. I was one among 19 awardees worldwide. I was also named as Khurana Scholar in 2019 and visited Massachusetts Institute of Technology in USA. During my stay at MIT, I worked on pathways regulating genome dosage sensitivity in endosperm. And I also wanted to see what are the other pathways regulating genome dosage sensitivity in Arabidopsis. My significant finding was that methylation required for dosage sensitivity is laid down in gametophyte rather than the. Also, we discovered the role of auxin in genome dosage regulation in plants. I have got 2 research papers accepted, one in Research Journal of Biotechnology and another in Indian Journal of Agriculture Sciences. I have also submitted sequences to NCBI through GenBank, and I have submitted 2 research papers, 1 book chapter and 1 review paper which are under review. I have participated in number of conferences and presented 4 posters and got 2 best poster awards. We went to zonal level in the ICR Kritagya Hackathon competition, pre-finals level in the National Bioentrepreneurship competition, and also first in quiz competitions at IRI. During COVID-19 pandemic, I was a volunteer for Global Consortium for Chemosensory Research regarding COVID-19 surveys. I am also part of a Next Generation Scientist Foundation of India. We are 25 members organizing internships opportunities for undergraduate students. My sincere thanks to ICR and IRI for funding, fellowships and all the opportunities. I would like to thank my advisor and co-advisor, professor and the Department of Plant Physiology and all my friends and family for their continuous support. I would like to rest my presentation here with a quote from John F. Kennedy. One who solves the problem of water is worth 2 Nobel Prizes, one for science and one for peace. Thank you very much.
Well, Prateek, that was very nice presentation. When you say a protein is disordered, what do you mean?
Sir, means the disordered structure of the protein don't have a definite structure like alpha helix or a beta sheet. So the alpha helix or a beta sheet is a stable structure, and the disordered structure is which we cannot model through various approaches, and the disordered structure is not having a definite shape. So it can assume any shape and enable the protein to bind to different proteins or the DNA.
The model that you showed did have beta sheets. I think it had also alpha helices 1 or 2 in the slide.
Yes, sir. Definitely, sir. Hello. Yes, sir. So I think my internet connection is a bit unstable, but yes, sir. Definitely, it has 2 alpha helices and 2 beta sheets running in parallel. And the other parts, it's disordered. So it's very small portion which is very ordered structure, and most of the structure is disordered. So that's what we have found.
Yeah. So it has ordered part, it has some disordered part. So calling it disordered totally, does that convey what you want to say, or what it is actually?
Actually, they normally the disordered protein term is coined for the proteins which has disordered structures in them, some part or the whole protein. But usually there won't be a whole protein disorder except for some viral capsid proteins. Okay. Usually in the plants or animals. Yes.
Yeah, you talked about CRISPR-Cas gene editing of this particular gene. Which part of the gene you are targeting and why?
Yes, sir. So initially we wanted to edit the gene. So we want to bring out mutations in the gene. And the efficiency of editing is usually higher at the 5' end of the gene. So what we have done is we have designed 2 guide RNAs as such to target the 5' end and 3' end, both the ends of the genes, so that we will have more chances of getting mutations.
You didn't get my point. You are expecting to inactivate this protein by mutating the 5' end of the gene. Is that what you are targeting, or you are targeting a specific part of the gene?
No, sir. We want to get the mutants. Just we want.
Well, I think we have lost him. We can take him up later on. You could tell him if he gets connected. Could we move on?
I think he must be trying. Just wait 5 minutes, sir. Krishnathan, or somebody can call him.
I'm calling him. Sir.
Hello.
Yes, Pratik.
Yes, Pratik.
Sir, very much sorry, sir. There is network issues.
You can keep your video off and then speak. Did Professor V.D. Singh had some question? Did you understand the question?
Yes, sir. Definitely, sir.
I think, sir, you need to repeat your question.
Yeah. Pratik, my question was this, that when you want to mutate a gene, you may have an objective of either inactivating it or doing something
Yes,
else,
sir.
like you expect some base substitutions in certain domains. Because even if it is non-homologous and joining, some percent of the mutations are because of base substitutions, not because of insertions and deletions.
Yes, sir.
Even 1/3 of insertions, deletions will be sense mutations. So what are you trying to do?
Correct,
Inactivate the gene or create some novel mutation?
We wanted to create novel mutations. Like, then we will screen for what is the effect of those mutations. So it's kind of generating random mutations and then screening for the effect of those mutations.
The amount of work you presented that you carried out.
Yes, sir. Yes, sir.
To me, it seemed quite large for an MSc student who had only less than 2 years to go. So
yes, sir.
You must have been working very hard.
Possibly, sir. I don't know.
Okay.
Sir, actually, yeah, I don't know.
Yeah. Other panel members, please.
Sir,
yeah, please.
Yeah, yes. Pratik.
Yes, ma'am.
Very nice work, and I just want to extend the question the chairman has asked regarding the development of mutants. So you yourself said that some part of the protein is organized and some part is not organized. You have alpha and beta structures. So while creating mutants, are you targeting some portion from the organized part also that you are taking care, or only the 5' 3'?
We are taking care of the 5' 3', ma'am, not the exact part, which is because we don't have any information about these proteins, which part is binding to DNA or something. This is something, the very preliminary studies we are doing, ma'am. Because after the studies from 2 Chinese labs in 2016, they just said that this is increasing expression during drought. So molecular aspect, we are the first discovering. So we don't have any information on which part is binding to DNA or something. So we could not target specifically which region to target. So we did try to create random mutation and then we will use the effect. That was the idea, ma'am.
No, it's good, but it's my suggestion that in your future work, because most of the organized structures like alpha and beta sheets, they are having some functional domains. So possibly they have role in function what you are looking at. So while designing different mutant, it is always good to have mutant in different parts, and some portion may be hitting the alpha helix or the beta structure. That is my suggestion in your future work.
Okay, ma'am. Thank you very much, ma'am.
Pratik, I have a question. If you want to make a very precise change in a gene, which approach you will use, SDN1, 2, or 3? Precisely what you want. You don't want to be any more random. You just want to induce the change that you want.
Yes, sir. Sir, can you repeat the question?
So if you want to make a very precise change that you want in a particular gene, which approach of whether you will use SDN1 or 2 or 3? of the three approaches of genome editing. Which one you will use?
SDN one. I think SDN. I am not clear, sir. Sorry.
You've not heard of SDN1, SDN2, SDN3? Site-directed nuclease 1, 2, and 3. Site-directed nuclease 1, 2, and 3. Who is your teacher?
Sir, Rakesh Pandey sir and Vishwanathan sir.
Nathan sir. Okay. All right. So fine. That's you made excellent presentation and excellent work done. So to be very precise, if you want to make a known change in a particular gene, you can see SDN1 is a random one. What you are trying to do, you may or may not get the change that you do. So you have to go for 2, and if you want to be very, very specific, you can still go for 3 when you want to bring from other places.
Okay, sir.
Thank you.
Thank you, sir. Thank you, sir.
Any other member? It seems the panelists have been convinced by what you presented. They have no questions. So excellent presentation, Pratik. Thank you very much. And we could move on to the next.
Thank you, sir.
Student.
Thank you, sir. I now call upon Soumya Sri A from the discipline of post-harvest technology.
Honorable Chairperson, respected jury members, distinguished scientists, and my dear friends, I welcome you all for my presentation. Horticulture has emerged as the main growth engine of Indian agriculture for the last 2 decades with spectacular performance in terms of production. During 2020 budget, our Finance Minister Nirmala Sitharaman had emphasized on horticulture production by implementing one horticulture crop in one district. India has witnessed a voluminous increase in horticulture production of 313.4 metric tons, but approximately 20% of the total horticulture produce go to waste owing to improper post-harvest management. In order to mitigate this, effective post-harvest management has to be taken up. Some of the methods to control post-harvest loss are hot water treatment, ionizing radiation, ozone treatment, chemical fungicides, biocontrol agents, edible coatings, and so on. But the thing is, not all the treatments are effective for all the fruits. For instance, some of the temperate stone fruits like apricot, peach, plum, nectarine are sensitive to chilling injury and are highly susceptible to post-harvest pathogens. Nowadays, many of the pathogens have developed resistance towards the use of chemical fungicides. So, there is need for a reliable and efficient method, and one such method is the use of edible coatings along with plant extracts, found to be the best. As this method is safe to use, easy to apply, eco-friendly, suppress microbial growth, increase shelf life, enhance physiochemical and organoleptic properties, cheap to access, reduce the risk of chemical residue, and increase marketability. Even though it has several advantages, but literature availability on these are very few. So the major research gaps are limited work on antimicrobial effect of plant extracts on post-harvest pathogens of nectarine, and no work on the impact of plant extracts along with edible coatings on decay control and shelf life extension of nectarine. With this background, we conducted a study entitled Post-Harvest Disease Management and Shelf Life Extension of Nectarine, and myself Soumya Sri from the Division of Food Science and Post-Harvest Technology. And the objectives of my research work are antimicrobial activity of plant extracts against major post-harvest pathogens of nectarine, and plant extracts and edible coatings for decay control and shelf life extension of nectarine. Nectarine Prunus persica variety Nuci persica belongs to family Rosaceae, is an important stone fruit crop after peach and plum, and it is considered as a natural mutant of peach which lacks fuzzy trichomes on fruit surface. Even though it has abundant of health benefits, but due to its juicy texture and high perishable in nature, it has very short shelf life of 3 to 4 days. And coming to objective 1, antimicrobial activity of plant extracts against major post-harvest pathogens of nectarine. To carry out my objective 1, 2 major post-harvest pathogens, that is Rhizopus stolonifer and Penicillium expansum, were identified, isolated from deceased fruits and subculture under lab condition. And methanolic plant extracts were prepared by using 5 different plant leaves like moringa, neem, marigold, ocimum, and eucalyptus. And antimicrobial activity of these plant extracts were tested against these 2 pathogens at 4 different concentrations like 0.05%, 0.1%, 0.2%, and 0.4% under in vitro conditions. In order to know the minimal inhibitory concentration, we followed 24-well culture plate method. By this method, we found that 0.2% concentration was the optimized. And interestingly, at this concentration, all the plant extracts had better control over sporulation. Further, to understand the combination effect, we prepared a mixed plant extracts by mixing all the plant extracts in equal proportion. And we found that mixed plant extract had better control over growth of pathogen as compared to plant extracts alone. After witnessing these outcomes, assessed colony diameter and inhibition percentage by using spread plate method. Colony diameter of rhizopus were confirmed after 48 hours of inoculation, and as compared to control, all the plant extracts had exhibited less colony diameter, but among the treatments, mixed plant extract exhibited highest antifungal activity. With respect to Penicillium expansum, the growth was confirmed after 72 hours of inoculation, and the colony diameter was comparatively less in mixed plant extract as compared to other treatments and control. And the inhibition percentage were highest with mixed plant extract at 0.2% concentration, followed by moringa, eucalyptus and marigold leaf extracts. So conclusions for the objective 1 are: methanolic plant extracts of moringa, eucalyptus, marigold, neem and ocimum had the potential to inhibit post-urge pathogens of nectarines effectively, and 0.2% was found to be the best concentration. And mixed plant extracts had better antimicrobial efficacy than using plant extracts alone. Coming to second objective, plant extracts and edible coatings are decay control and shelf life extension of nectarine. To accomplish my second objective, commercially grown snow queen nectarines were selected and coated with 8 different treatments like mixed plant extracts alone 2%, chitosan alone 1%, carboxymethyl cellulose alone 1.5%. And by using these 3 treatments, layer by layer coating was carried out by dipping the fruits in one treatment for 10 minutes, air dried for 10 to 15 minutes, and followed by dipping in another coating for 10 minutes and air dried for 10 to 15 minutes. And finally stored at 2 different storage temperature conditions like supermarket conditions for 18 plus or minus 2 degrees Celsius, 85 to 90% storage for 15 days, and another at cold storage condition 1 plus or minus 1 degree Celsius, 85 to 90% storage for 30 days. And during this experiment, treated and untreated fruits were evaluated for physical, physiological and biochemical attributes at 5 days interval for cold store fruits and at 4 days interval for the fruits stored at supermarket conditions. And coming to the result part, physiological loss in weight was comparatively less in CMC, CH and MPE treated fruits, as it exhibited only 9% loss as compared to other treatments and control fruits during supermarket condition as well as cold storage condition. With respect to respiration rate, control fruits had its respiratory peak at 8th day under supermarket condition and at 15th day under cold storage condition, while CMC, CH and MPE had its respiratory peak at 16th day under supermarket conditions and at 25th day under cold storage condition. This implies that deterioration rate was faster in control fruits as compared to treated fruits. Coming to fruit firmness, fruits coated with layer by layer coating of CMC, CH and MP had maintained a constant firmness throughout the storage, while in case of control fruits, fruit firmness decreases gradually. Coming to biochemical attributes, fruits coated with layer by layer coating of CMC, CH and MP maintain highest ascorbic acid content, total phenolics and antioxidant activity as compared to control under both supermarket condition as well as cold storage condition. With respect to sensory attributes, the overall acceptability score was good for all the treatments. During supermarket conditions, the control fruits were unacceptable on 8th day of storage, while CMC, CH and MP treated fruits were acceptable even up to 16th day of storage. And with respect to cold storage, control fruits were unacceptable on 15th day of storage, while CMC, CH and MP treated fruits were acceptable up to 25th day of storage. So conclusions for the second objective are: layer by layer coating of CMC, CH and MP proved to be the best, and as it extends the shelf life of nectarines up to 16 days under supermarket conditions and 25 days under cold storage conditions. So silent findings of my research are: the antifungal activity of mixed methanolic plant leaf extracts was prominent in controlling the growth of Rhizopus stolonifer and Penicillium expansum, and hydrocolloid based coatings alone or in combination with mixed plant extract reduce fruit decay and weight loss and maintain the functional attributes of nectarine fruits during the storage period. However, layer by layer coatings of CMC, CH and MP was the most effective approach. Further, the research can be extended to evaluate the antimicrobial efficacy of plant extracts on different pathogens causing post-urge spoilage, and need to identify the compounds responsible for antimicrobial activity in different plant extracts. I have attended many national conferences and webinars, and I awarded with Best Poster Award during National Conference on Food Safety, Nutritional Security and Sustainability. From my research work, one paper has been accepted in Indian Journal of Agriculture Sciences with the NAS rating of 6.25, and other 2 papers are under review in the Journal of Food Processing and Preservation and Octophysiology Plant. I duly acknowledge my chairperson, Dr. R.R. Sharma, members of my advisory committee, faculty of Division of Food Science and Post-Storage Technology, Dean PG School IRI, ICR for Junior Research Fellowship, parents, teachers and my friends. By this, I would like to end up my presentation with the saying of E.O. Wilson, nature has the power to heal our pain, also power to ruin. The results vary on how we treat Mother Nature. Save nature, save earth and save your lives. Thank you one and all.
Thank you, Soumya. Now it is open for discussion. Probably during your PowerPoint presentation, there was a lot of sound in the background.
Yes, sir.
I would like one clarification. There was some disturbance during the presentation of my internet connection or whatever. I just want some clarification. You are mentioning 2 conditions: supermarket conditions and cold storage conditions.
Yes, sir. Supermarket.
Yeah, what is the
Yes,
difference
sir.
between these 2?
Sir, supermarket condition is nothing but the stores where we can purchase. It is a normal shop service, sir, where we can purchase the fruits. But cold storage is nothing but it is a storage room where we can, after harvest, we can store the fruits.
Temperature.
No, I mean temperature differences.
Temperature difference is there, sir.
How much?
Tell. Sir, in cold storage we maintain 0 plus or minus 2 degrees Celsius, and in supermarket condition we maintain 18 plus or minus 2 degrees Celsius.
Okay, thank you. So this increases the shelf life by 16 days, you mentioned?
16 days. Yes, sir, 16 days, sir.
Okay.
In supermarket condition, and in 25th day under cold storage condition.
Shri, I have 1 or 2 questions.
Okay, sir.
These plant extracts coating, do they come under FSSAI regulations and guidelines?
Yes, sir. They are generally regarded as safe, sir. These plant extracts are possessing antimicrobial activity, sir. And see what the treatments I have used, that is CMC and chitosan, are regarded as safe,
So
sir.
they fall under FSSAI
Yes, sir.
the regulations and guidelines. If
Yes,
that
sir.
is the thing, what further? When you say path ahead, you should tell about those things also.
Okay, sir.
Safety is very important thing. Okay.
Okay, sir.
Also, when you use plant extracts, don't you think that the material which used for extraction
Yes,
will
sir.
be there along with the extract? Will they not cause any practical problem? Because you have to now take it up further in
Yes,
terms
sir.
of its practical use.
Yes,
So
sir.
how is that you are going to interface this one with the practical application?
Sir, we have used methanolic plant extract, sir. Methanol as a solvent. Before we apply on the fruit, we evaporate the methanol content, sir, by using rotary evaporator, and then we mix the components, plant components which are dried, partially dried, with phosphate buffer to maintain the pH. So it is not affect for the health.
I am asking you how it is practically feasible in a situation where you want to use them as coating. So because you have to coat them individually, or how it is to be done. So some kind of application methodology is to be developed, is it not? Large scale, mass scale or large scale methodology.
Yes, sir. This is followed for commercial scale, sir.
Yes. Okay. Okay. Otherwise, good work. Okay.
Okay, sir.
Soumya,
Yes, sir.
I compliment for your eco-friendly approach
Yes, sir.
of
Eco-friendly.
edible coatings and plant extracts
Yes, sir.
for increasing the shelf life
Yes, sir.
of nectarine, which is very important
Yes,
fruit.
sir. It is very rich in antioxidant and vitamin
So-
C, which boost immune system, sir.
So you used, marigold, neem, and
Osimum.
also moringa.
Yes, sir.
What
Moringa.
are the phytochemical compounds?
Sir, the study has to be taken out for this analysis, sir. During my research work, the gas chromatography machine was not working, sir. So I dropped this. Further, I have to take up, sir.
And marigold, which is the species used?
French marigold, sir. That is Tagetes spatula.
French marigold. Okay.
Yes, sir. French marigold.
So these are all leaf extracts used?
Leaf extracts, sir. Leaf.
Because moringa, even pods.
Yes, sir. We can use seeds, pods, and the roots also we can use, sir.
Good work.
Thank you, sir.
Rameshri,
Yes, sir.
what is the stability of the plant extract? How long the compound should be stable?
Sir, so that's why we have taken methanolic solvent, sir. Means it is stable for more days, sir. Methanol extracts the phytochemicals, all the phytochemicals present in the leaves, sir, and it is stable for more time. That's why we use methanol solvent as compared to aqueous and other solvents.
What was the basis for taking up mixed plant solvents, mixed plant extracts?
Yes, sir. Sir, when we observed that at 0.2% concentration, all the plant extracts had better control over sporulation, sir. So for our curiosity, we mixed all the plant extract, and we tried on the growth of pathogen, and it showed very good result as compared to plant extracts alone, sir. And these plant extracts are easily available in our near surroundings, sir.
No, what was the reason for coating with carboxymethyl cellulose and all that?
Okay, sir. Actually, carboxymethyl cellulose is a very good addition, sir. As compared to chitosan, it is very good addition, and it will stabilize the internal layer, sir. We used as an internal layer. It will provide barrier to moistureless and gas, sir. That why. And we used chitosan because it has an antimicrobial property, and it imparts glossiness to the fruit. And mixed plant extracts fight against the growth of fungus.
So it is the cumulative effect of all
Yes,
this
sir.
put together and the shelf life.
Yes, sir.
By what time you can really extend the shelf life?
Sir?
What is the time you can extend?
Sir,
By what time you can extend the shelf life of the food?
In supermarket condition, we can extend up to 16 days, sir. And in cold storage condition, we can extend up to 25 days. But in control means without any coating, that fruits were they are perishable within 3 to 4 days, sir. We can extend up to more than 10 days and 15 days.
Thank you.
Thank you, sir. Thank you, sir.
Soumya, just calling you to hypothesize.
Okay, sir.
4 plant extracts individually, they are effective, but when they are mixed,
yes, sir,
the mixed extract is much more effective than individuals.
Yes, sir.
Instead of 0.2, I think you needed 0.05% only for
0.05, 0.1, 0.2, and 0.4, sir. At all the concentration in mixed plant extracts, at all the concentration, there is less growth, sir. But at 0.2% concentration, there was complete inhibition of the pathogen, sir.
Okay. So why should mixed plant extract be more effective than individual plant extracts?
Yes, sir.
Just a hypothesis.
Okay, sir. As compared to using plant extracts alone, when we mix all the plant extracts, it will give additional synergetic effect against the growth of pathogens, sir. Means synergetic effect.
How? Why this can happen? Any possibility?
Okay, sir. Because all the plant extracts have different secondary metabolites, sir. For example, in marigold, some of the terpenes and all there, sir. And moringa, it has some another antimicrobial and antifungal activities, sir. When we mix all these plant extracts, all the secondary metabolites act upon the growth of pathogens. Means that will inhibit the growth.
Okay. Professor Jha, we can thank the candidate.
Thank you, Soumya Sri. Thank you, sir. And we move ahead. And next contestant is Sahil Kumar from Seed Science and Technology. Sahil Kumar.
A very warm welcome to you all. I am Sahil Kumar from the Division of Seed Science and Technology. Let me begin with a quote in Manusmriti, Subijam Suchetre Jaite Sampadate, meaning good seed in good soil always give better yield. Rice is the staple food in many parts of the world and India. Ever-growing population and decreasing agriculture resources may lead to food scarcity in near future. Hence, to maintain the sustainable rice production, persistent emphasis has been given for rice crop improvement since ages. That resulted in release of large number of varieties with improved yield, quality, tolerance to biotic and abiotic stresses. This leads to a Herculean task to maintain the seed quality. The seed quality attributes comprises of physical purity, genetic purity, germination, seed health, moisture, and seed vigor. Although all these attributes are equally essential, but the purity components require our intent. So how seed impurities occur? It occurs during seed multiplication, processing, transportation, storage, and because of natural crossing and genetic variations. So what is the way out? Assessment of seed impurities is based on morphological characters which are season specific, take longer time, and are not fully accurate. In contrast, chemical methods require some technical expertise and are distinct, easy, and give reproducible results. Here comes the phenol test. It is based on polyphenol oxidase activity. It involves hydroxylation of aromatic ring of phenol to form L-dopa and catechol in presence of tyrosinase and oxygen, and these catechol undergo oxidation to form quinones, and polymerization of quinones leads to melanin formation. The reaction is controlled by tyrosinase activity in seed coat, which is under genetic control. Phenol tests help in characterization and identification of plant varieties in crops like wheat, rice, and coffee. It helps breeder to assess the diversity of breeding stock, seed testing authorities to test genetic purity and certifying a seed lot, registration authorities to protect a plant variety under PVP Act, and marketing personnel to certify grains for consumer protection. Realizing this, gloom phenol reaction has been included as one of the characters for DOS testing under UPOV and PPVFRA India. However, based on earlier experiments conducted, a clear-cut grouping of varieties based on phenol color reaction couldn't be obtained. Only two groups, namely present and absent, was considered in DOS testing system. This prompted us to search for the cause of variation in phenol color reaction in rice varieties and its biochemical basis. Further, a literature on phenol test methodology followed by researchers is variable and it is not recommended by International Seed Testing Association. With this background, we formulated our research on an essay on phenol color reaction in rice seed. Objectives set were to standardize the methodology for phenol color reaction, to assess the source of variation in varieties for phenol color reaction in relation to growth and maturity of seeds, to estimate different polyphenol oxidases in relation to phenol color, and to establish a relationship between age of seed and phenol color reaction. In first objective, 5 varieties, one each of different phenol color group, were used. 50 seeds, each in 2 replication, were soaked in distilled water for 16 hours at room temperature. Petri plates of diameter 9 and 11 cm with 4 and 5 ml of phenol solution at concentration 1, 2, 3 and 4% were used. Petri plates were kept in an incubator at 25 degrees Celsius in dark, and color development was recorded at interval of 2, 4, 6 and 8 hours. Results showed that color development varied at different time intervals. The stable color was observed at 4 hours with 2% phenol concentration either in 9 cm Petri plate with 4 ml or 11 cm Petri plate with 5 ml phenol solution in phenol color groups. In second objective, to assess the source of variation, 60 released varieties were used which were grown in Kharif 2019. All the varieties were grown and sampling were done at different stages of growth like pre-anthesis, 10, 20 and 30 days after anthesis, and tested for phenol color reaction. Results showed that phenol color reaction did not occur in unfertilized spikelet, but phenol color reaction developed at all the stages after anthesis. To study interpanicle variation, 3 panicles in each variety harvested at maturity, namely main tiller, mid tiller, and late tiller, and phenol color reaction was recorded, which reveals no significant difference across the phenol color groups. Similarly, to assess the intrapanicle variation, panicles were sampled and divided in 3 parts: top, middle, and bottom, and phenol color reaction was observed. Slight variation due to position of seed in panicle was found. In third objective, rice husk powder was mixed with methanol and kept in an incubator shaker at 37 degrees Celsius with 300 rpm for 4 hours. Extract of phenolic compounds were mixed with Follin's reagent followed by sodium carbonate, and absorbance was taken after 2 hours of incubation. Results showed that highest phenol content was found in second stage and decreased thereafter. Interpanicle phenol content was not significantly different, whereas intrapanicle difference in phenol content was observed. Polyphenol oxidase activity was estimated by taking phenolic substrate tyrosine, pyracatechol, and L-DOPA in 2 ml centrifuge individually, and 5 rice seeds were added to that, followed by incubation at room temperature with rotation for 30 minutes, and absorbance were taken. Results showed that polyphenol oxidase activity in relation to phenol content in all color groups. In fourth objective, seeds were stored in ambient condition for 1 year, followed by screening for phenol color percentage, phenol content, and polyphenol oxidase activity. Results showed that stored seed lots have higher intensity of phenol color in seeds, higher phenol content, and increased polyphenol oxidase activity like L-DOPA, tyrosine, and pyracatechol. The overall conclusion of our research work is methods for phenol reaction test in rice seed standardized, source of phenol color variation in fresh and stored seed identified, biochemical basis of phenol color reaction found. Our research work can further be forwarded as standardized methodology can be used for quick estimation of varietal admixture in seed lot, can be used in varietal identification and registration system. 5 groups recommended instead of present and absent based on quantitative analysis of polyphenol oxidase. Suitable rapid testing kit can be developed. 2 research papers have been accepted and 1 is under review. 1 abstract published got best poster presentation for my work, got prize in Hindi Shruti Lekh. I have participated in various cultural events and attended training for 10 days at Karnal. I would like to acknowledge everyone who helped me directly or indirectly. I end my presentation with this beautiful quote given by William A. Foster: "Quality is never an accident. It is always the result of high intention, sincere effort, intelligent direction, and skillful execution." Thank you.
Thank you, Sahil. Very well presented. Open for discussion, sir.
Well, Sahil, that was very practically oriented work. Can you explain why there should be intrapanicle variation in phenol reaction?
Yes, sir. The maturation in panicles start from the middle and extend towards the top and bottom. So it takes time to mature the panicle fully. So it takes around 7 to 8 days to mature fully. So there is a difference in the phenol content and color percentage at the bottom of the panicle.
I thought you had used mature panicle, mature at the maturity stage, but it was perhaps much before physiological maturity stage.
Yes.
Okay. So what would you advise people who are involved in testing, seed testing, to be careful about phenol test?
Sir, the period of soaking in phenol concentration during incubation, sir. Actually, previously, only 2 groups, present and absent, was there. So it was based on like only 2 color groups, present and absent. So now it is extended towards 5 color groups, different color group like dark brown, black, and those things. So incubation is up to 4 hours. If we incubate for larger period of time, then it gives some, like earlier they used to soak for incubate for in phenol 24 hours or more. So there is a somewhat color variation. So they should be careful about the fresh seed and stored seed, because there is a variation in a stored seed. We are seeing the increase in phenol content and phenol color intensity of the phenol color percentage while we are determining the phenol color reaction qualitatively.
So essentially two things: the seed maturity is one issue, and the storage period is the other issue. And then of course incubation period, that is the third issue. That should be standardized.
Yes, sir.
Okay. Other members, please.
Ayel?
Yes, sir.
I have a question to you. You have done it on rice, is it not?
Yes, sir.
Is there any other crop to which this methodology can be extended?
Yes, sir. In wheat, it is standardized, sir, by and it is under approval recommendation of International Seed Testing Association.
Okay. And what about rice? It is not been still done?
No, sir, it is not done, sir. Under International Seed Testing Association, it is not recommended, sir, for purity testing.
Okay, this is what I want to know. Thank you.
Thank you.
So Sahil, you are talking about two variables. One is phenol content and another is polyphenol oxidase activity.
Yes.
So according to your observation, which is more critical for color development?
Polyphenol oxidase activity, because the color development, melanin formation is a key index of polyphenol oxidase activity.
Okay, so you are planning to develop a kit. So
Yes,
you
sir.
have to do it in the form of a kinetics, that at what concentration the polyphenol oxidase is giving you a color.
Yes, ma'am.
So that is your main focus.
One
And
of the
phenol concentration is not playing that role?
Yes, ma'am, it is playing. But if you see in comparison basis, then PPO activity is more means it is important than phenol content.
Okay. Okay.
Thank you.
Seems no. Yeah, please.
Chairman, sir, can we go ahead?
Yeah, please. That is what I was going to say.
Thank you, sir. And thank you, Sahil. Now, I invite Suri Prakash Yadav from the discipline of Soil Science and Agricultural Chemistry.
Very good afternoon to one and all present here. I shall start my presentation with a quote of H.H. Pinnett: "Out of the long list of nature's gifts to man, none is perhaps so utterly essential to human life as soil." Why I quoted here, let me go before Green Revolution era, where food insufficiency and famine was common word among policymakers, scientists, producers, as well as consumers. That led a major segment of our population to suffer from starvation and malnutrition. At the same time, we thank to Professor M.S. Swaminathan for his vision. Today, India has become exporter of several agricultural producers. The major driving factor behind Green Revolution was high-yielding varieties, chemical fertilizer, and irrigation. However, the burden for exponentially increasing population led to overexploitation and further degradation of natural resources. Increased production and productivity in post-Green Revolution era was achieved through intensive agriculture, but on the other side, it led to deterioration in soil health because of depletion in organic matter in soil, nutrient mining, and soil erosion. Moreover, intensive agriculture also led to decline in factor productivity and increased level of carbon dioxide into the environment. Today, crop residue burning, climate change, and decrease in water table is a buzzword among policymakers and scientists. These burning issues severely distressed our society and way of living. The government of India estimated around 11.3 million tons of residue was burned in the year of 2019 by 2 Indian states. During crop residue burning, it emits large amount of greenhouse gases that contributes to global warming and severely affect our health. Besides that, crop residue burning is detrimental for soil flora and fauna that are engaged in the transformation of nutrient and biogeochemical cycle. In monetary terms, it's a huge loss, which is equivalent to 1.15% of our GDP. Now condition is worsening to the bottom level when even government and other apex bodies remain clueless to handle the situation. Then what could be the probable steps to be taken for restoring and improving degraded soil health caused by conventional intensive agriculture? CA has a potential to tackle the alarming situation aroused due to intensive agriculture, which is based upon these principles. A lot has been talked about positive impact of conservation agriculture on soil organic carbon. Then question arises, what about the most important primary nutrient, that is nitrogen? Still, its transformation under conservation agriculture is in enigmatic stage. In this background, we have carried out the research work entitled Soil Nitrogen Transformation in Pigeon Pea and Cotton under Conservation Agriculture. And the objectives were to study soil nitrogen release and microbial function in pigeon pea and cotton under conservation agriculture, and another was to study the impact of conservation agriculture practices on soil nitrogen fractions in pigeon pea and cotton. We had chosen weed-based leguminous and non-leguminous cropping system as their contribution is significant in food security and economic stability of the Asian countries. To achieve these objectives, soil sample was collected from 10-year-old conservation agriculture based experiment at ICR IRI New Delhi. The treatment include conventional tillage plots with flat bed, while rest of the treatment include permanent narrow bed, permanent broad bed, flat bed with and without residue retention, along with zero tillage with similar dose of fertilizer in each plot. The soil sample was collected at 4 critical crop growth stage of pigeon pea and cotton from 3 different depths. The initial properties of soil are as follows. Under first objective, we analyzed these soil properties. The determination of ammonium and nitrate were performed as per the procedure of Keeney and Nelson, and the extract were quantified on flow injector analyzer. For arginine deaminase activity and nitrogen mineralization potential, the sample was incubated and further analyzed through FIA-FOS. Coming to result, continuous crop residue retention along with zero tillage significantly enhanced ammonium nitrogen concentration at different crop growth stage in 0 to 5 cm of soil depth. Among residue retained plot, permanent broadbed was superior over permanent narrow bed and conventional tillage plots at different crop growth stages. Lower concentration of ammonium was recorded at deeper soil, although the treatment effect was similar as like 0 to 5 cm. Whereas in cotton, similar result was observed as like under pigeon pea crop, except the concentration of ammonium was lower as compared to pigeon pea. Higher concentration of ammonia in residue retained plot with zero tillage might be due to stimulation of ammonification process by crop residue. As like ammonia concentration, nitrate concentration in soil was also higher in residue retained plots as compared to conventional tillage plot. In 0 to 5 cm of soil depth, pot filling stage of pigeon pea recorded higher nitrate concentration, whereas at 5 to 15 cm of soil depth, pre-flowering stage had higher nitrate concentration in soil. In cotton, maximum nitrate concentration was recorded at ball formation stage in both the soil depth, and the treatment effect was similar as in pigeon pea crop. The higher concentration of ammonia and nitrate in pigeon pea wheat cropping system might be due to nitrogen fixing ability and higher root biomass of pigeon pea as compared to cotton. Arginine deaminase activity signifies the mineralization of soil organic matter. Among crop growth stages, highest activity was noticed at pot filling stage in both the soil depth. Among residue retained plots, permanent broadbed had higher arginine deaminase activity. In 5 to 15 cm of soil depth, residue retained permanent broadbed plot had almost 2 times higher arginine deaminase activity than conventional tillage plot, irrespective of growth stage. In cotton crop, impulsive increase in arginine deaminase activity was noticed at ball formation stage from flowering stage. Lower activity at flowering stage depict the inhibitory effect of ammonium nitrogen and concentration. Soil nitrogen mineralization potential is directly linked with availability of nitrogen in soil plant continuum. We found that residue retained broadbed plot had 80 to 120% higher nitrogen mineralization potential of soil than conventional tillage plot in 0 to 5 cm, whereas the same was varied between 50 to 110% in 5 to 15 cm of soil depth. Highest soil mineralization potential was recorded at flowering stage of pigeon pea. Further steep reduction in nitrogen mineralization potential was noticed at pot filling stage. Coming to dehydrogenase activity, which depict overall microbial activity of soil, it was noticed that conservation agriculture based plot had 45 to 120% higher dehydrogenase activity than conventional tillage plots in 0 to 5 cm of soil depth. Almost similar trend was noticed in 5 to 15 cm of soil depth. As like pigeon pea, higher dehydrogenase activity was noticed in residue retained plots than conventional tillage plots, irrespective of crop growth stage and soil depth. This might be associated with higher substrate availability and congenial soil conditions in residue retained plots. For the second objective, soil samples were collected at harvesting stage of crop growth in 0 to 15 cm of soil depth in both the crops and determined total nitrogen and other hydrolyzable fractions of soil. For hydrolyzable fraction, sample was digested 12 hours and different fractions were quantified as per procedure suggested by Bremner. Crop residue retention substantially improved total nitrogen in soil. Under residue retained plots, 33 to 39% higher total nitrogen was noticed as compared to conventional tillage plots, whereas in cotton crop under CA plots had 19 to 28% higher total nitrogen as compared to conventional tillage plots. Hydrolyzable nitrogen constitutes 74 to 90% of total nitrogen in soil under pigeon pea crop. Most importantly, residue retention along with zero tillage increased hydrolyzable nitrogen fractions in soil as compared to without residue retained and conventional tillage plots. Further, hydrolyzable ammonia nitrogen constitutes the major fraction of hydrolyzable nitrogen and it varied between 24 to 37% among treatments. Hydrolyzable ammonia nitrogen considered as easily available pool of nitrogen for microbes and plants. Another important fractions of hydrolyzable nitrogen, that is amino acid nitrogen, which depict the long-term nitrogen supplying capacity of soil, was in the range of 47 to 50% of hydrolyzable nitrogen among CA plots. Hydrolyzable nitrogen in soil under cotton crop was varied between 77 to 88% of total nitrogen and it was highest under flatbed plus residue retained plot. As like in pigeon pea crop, hydrolyzable ammonia nitrogen and amino acid nitrogen was the major fraction of hydrolyzable nitrogen and it was positively affected by crop establishment method and tillage practices. In conclusion, among the treatments, permanent broadbed plus residue retention proved to be a better and a viable soil management option for enhancing nitrogen supplying capacity of soil and soil biological functions. Substantial improvement in microbial function, total nitrogen, hydrolyzable ammonia nitrogen as well as amino acid nitrogen under CA practice depict its long-term sustainability in pigeon pea wheat and cotton wheat cropping system under semi-arid climate. For better understanding, community composition profiling and gene-based study is required to understand the driving force behind changes in soil function and nitrogen transformation in rhizosphere and non-rhizosphere. From these research findings, 2 research papers have been accepted in Indian Journal of Agriculture Science with NAS rating of 6.25. From our research work, I have presented 1 oral and 1 poster presentation. I have participated in different workshops and awarded with certificates. At the flag end of my presentation, I sincerely acknowledge my advisory committee and all of them who directly and indirectly helped me in my research as well as in academics. Finally, I would like to wrap up my presentation with a quote, "Take care of the land and the land will take care of you." Thank you all for your kind patience.
Professor Ramaswamy, would you like to take over?
Prakash.
Yes, sir.
Yeah, with the permission of chair, Surya Prakash.
Yes, sir.
Aspargine and glutamine are supposed to be 2 amino acids which constitute almost 20 to 26% of organic nitrogen.
Yes, sir.
Probably, did you do any work related to transformations of these 2 amino acids by enzyme acids?
No, sir. I did not did that. I'm looking forward in PhD because of time limitation. I did some part of that.
Okay.
And for extra, I will do in future.
Okay. This dehydrogenase activity, you could get maximum activity around flowering or pod formation stage. Could you tell me the reasons why you could get enhanced reactivity at the particular stage?
Yes, sir. Because of the rhizo deposition that are maximum at that stage. Because of that, carbon availability and for microbes is maximum. That is why the dehydrogenase activity at pod filling stage and near flowering stage was high.
Did you take it near the rhizosphere or in bulk soil? Did you analyze the rhizosphere? Okay. Thank you.
Yes,
Thank
sir.
you.
Thank you, sir.
Is there any rationale for taking
Yes, sir.
pigeon pea and cotton? Why did you take up these 2 crops? What is the reason? Why this combination also?
Sir, Sir, these two crops are contrasting. One is leguminous based and one is non-leguminous based. So I want to check whether the transformation is affected by taking legume crop or not.
Okay, leguminous and non. What
Yes,
will happen to PGNP, especially when it is grown as a perennial? Because in most of some of the northeastern parts of the states, it's being grown as a perennial crop, PGNP.
Yes, sir.
So, with your results, what can you forecast for them?
Sir, in my case, the variety was Pusa RR16. It's a short, extra short duration variety, and the crop root biomass in extra short variety is less as compared to the perennial root biomass. So, in that case, sir, can you repeat the question once again?
What will you can extend the results to a perennial situation like that of what is existing PGNP grown in northeastern parts of the country?
Sir,
because when it is grown as a perennial, the biomass and residues are totally different from when it is grown as an annual. Okay, that's it.
Yes, sir. Yes, sir. Sir, for that, research on ground level, because our result is on the basis of short duration variety. For long duration variety, we have to check whether the transformation is same or not. We can recommend, but we have to check on ground level, sir.
Okay.
So between the two crops,
yes, ma'am.
Where did you find the better observations in terms of your soil nutrients as well as microbial activities?
Ma'am, in case of pigeon pea weed cropping system, I get the better result because of the nitrogen fixing ability of pigeon pea roots, and the microbial activity was also higher in that cropping system as compared to cotton wheat cropping
As
system.
far as nitrogen fixing ability of pigeon pea, you are presuming, or did you actually see any nodulation in the pigeon pea?
No, ma'am, I did not see any nodulation and all these things. But on behalf of microbial parameter, what I analyzed, on that basis, I am saying.
Yeah. Thank you.
Thank you, ma'am.
When you said dehydrogenase activity, what dehydrogenase you are trying to refer to?
Actually, sir, it depicts the overall microbial activity in soil.
Okay, how do you measure it?
By incubating the soil sample at 28 degrees centigrade, and after that, I have analyzed on a spectrophotometer.
What is the substrate that you provide? Do you provide any substrate or only soil
Yes, sir.
substrate?
Yes, sir. No, I have provided a substrate. It's TTC, sir. Triphenyl tetrazoline chloride.
Okay.
So yeah, you have to actually tell them how this dehydrogenase activity works, the enzyme. What is the substrate and what is the product?
Actually, ma'am, the TTC works as electron acceptor, and after incubation, it's in the form of triphenyl formazone converted, which we take the reading after formation. We extract with the methanol. The TFF is extracted with methanol.
So, sir, it's basically like a respiration activity and it is applicable for all the microbes. It's not specific for any bacteria or fungus or anything.
Yeah.
It's an overall microbial activity, and it depicts the metabolic activity of the soil.
Yeah. So, Professor Jha, I think there are no further questions, it seems.
Thank you, sir. So we move ahead. I now invite Ms. Rakshita KN from Division of Vegetable Science.
A pleasant good afternoon to all the dignitaries, jury members on and off of the dais, and audience. I welcome you all. I hope all are doing good and safe. I would like to start my presentation with a famous quote by Albert Einstein: "Nothing will benefit human health and increases the chances of survival of life on the earth as much as the evolution to the vegetarian diet." Changing climate is going to be a big problem. It is happening here and everywhere now, and it leads to the global warming. No doubt it also affecting the plant growth and development. Production of cauliflower is hampered because of stresses. It is of two types, biotic and abiotic stresses. Stepping to abiotic stress, it is due to drought, salinity, heat, light, excess or deficiency of nutrient, heavy metals and pollutants. Cauliflower is highly nutritious, which is rich in phosphorus, protein, potassium, glucosinolates, anthocyanin, beta-carotene and antioxidants. Many researchers have developed purple and orange cauliflower in order to increase the nutritional quality of cauliflower. It is precisely the cauliflower has been grouped into four types: early, mid-early, mid-late and late, based on the specific temperature requirement for curd initiation and development, and any deviation in this required temperature may cause a huge economic loss to the farmer and causes many disorders like riciness, pink coloration, fuzziness, buttoning, no curding as well as leafiness. And many researchers have conducted an experiment to study the effect of temperature on curd initiation and curd development, identified the QTL, but this is not in case of Indian cauliflower. The main research gap is that there is a need to study the amount of diversity present in Indian cauliflower for curding traits related to temperature and understanding these curding traits by using molecular tools and to breed the varieties which is having a better plasticity related to temperature factor and mapping for temperature dependent variation, which was done in European cauliflower but not in case of Indian cauliflower. Mapping, it is of two types, genetic mapping and physical mapping. And genetic mapping is again classified into two types, QTL or biparental mapping and association mapping. In case of biparental mapping, the name itself saying that it involves only two parent and only one kind of recombination at a time. But in case of association mapping, it is taking the advantage of linkage disequilibrium to link the phenotype to genotype. To this background, we postulated our thesis title, Association Mapping of Heat Stress Related Curd Trait in Indian Cauliflower, myself Rakshita from Vegetable Science Division. To the help of my advisory committee, we postulated two objectives: to assess the phenotypic variation for temperature dependent curding traits in Indian cauliflower, and objective 2 is to analyze the molecular diversity in cauliflower germplasm and find their association with curding traits by using genotyping by sequencing. We selected 96 genotypes, in which 91 from all maturity groups and 1 from specialty type orange cauliflower and 2 from each broccoli and tropical cabbage. These 96 genotype has been grown at two locations, that is in IRI New Delhi and ICR Research Complex for Northeastern Hilly region, Barapani. We recorded a detail of temperature from sowing to harvesting at Delhi location as well as in Barapani location. We recorded morphological need related traits such as days to 50% curd initiation, curd harvest, curd length, curd diameter, marketable curd weight and net curd weight. Stepping to the results, the earliest curd initiation and curd harvest has been recorded by the genotype Pusa Meghna at Delhi condition and CC15 at Barapani condition. And the curd length and curd width has been recorded by KT25 and KT22 at Delhi condition and DC351AA at Barapani condition. The maximum marketable curd weight and net curd weight has been recorded by genotype KT20 at Delhi condition and BR2 at Barapani location. Genetic parameter for agromorphological traits showed that highest PCV and GCV was highest for net curd weight and heritability was highest for days to 50% curd initiation. These results are in line with the Barapani centers also. The clustering of genotypes based on D-square analysis showed that grouped the 96 genotype into 10 clusters and 7 clusters at Delhi and Barapani centers, in which cluster 7 has maximum genotypes and cluster 3 has maximum genotypes at Delhi and Barapani center respectively. The genotypic correlation study showed that the marketable curd weight is highly significant and positively correlated with net curd weight, followed by days to 50% curd initiation with the days to 50% curd maturity in Delhi and Barapani condition. The dendrogram depicting the 96 genotype into two main clusters, and again the main clusters are divided into subclusters. The salient findings from objective 1 is that the early group genotypes recorded less yield and late group genotypes took maximum days for the curd initiation and have recorded very good yield and good quality. The early group genotypes took mean value of 56 days and 30 days for curd initiation in Delhi and Barapani center respectively and high tolerance to temperature, and the late group took maximum days of 62 days in Delhi condition and 33 days for Barapani center for curd initiation, showing a very sensitivity to the temperature. The highest marketable curd weight and net curd weight was recorded by Snowball Group, and it took the maximum days for vegetative growth and curd development. And stepping to the objective 2, to analyze the molecular diversity in cauliflower germplasm and find their association with curding traits by using genotyping by sequencing. A detailed phenotyping of 96 genotype, the molecular diversity analysis was done by using 90 SSR marker. Out of 90 primers, 59 were polymorphic, having a major allele frequency of 0.5 to 1, and number of alleles was ranged from 1 to 9, having a genetic diversity ranged from 0 to 0.73, and polymorphic information content was ranged from 0.1 to 0.68. The structure analysis showed 96 genotype into 4 subpopulations. From the results of GBS, approximately 302 million reads in the 96 cauliflower genotypes with an average value of 3.1 million reads. This is the distribution of sequencing data for each sample after filtering and mapping ratios of generated data. We detected approximately 35,831 SNPs, in which chromosome number 4 has maximum SNPs and the minimum SNPs observed on chromosome number 6, having an average value of 80 SNP per MB. This is the distribution of SNP in chromosomes, and all these occurred within the centromeric and pericentromeric region. The major allele frequency in Indian cauliflower was ranged from 0.4 to 0.9, and the minor allele frequency was ranged from 0.02 to 0.5. The linkage disequilibrium decay varied across the different chromosomes, having from 0.25 to 0.33, with an average linkage disequilibrium having a value of 0.045. Among transition and transversion, the percent of transmission was higher than the percent of transversions, in which 63% and 36% respectively. Among transition, the CT transmission was higher, followed by AG transition, and among transversion, AC transversion was followed by GT, AT, and CG transversion. This is the dendrogram that showing the genetic relatedness among the 96 genotype by using 35,381 SNPs, and this is the population structure in cauliflower diversity divided the 96 genotype into 4 subpopulations. We detected 121 SNPs, in which 38 from Delhi region and 83 from Barapani conditions, and among Delhi condition, days to 50% curd initiation and curd harvest as maximum number of SNPs. Among Barapani center, marketable curd weight and net curd weight as maximum number of SNPs, out of which 25 SNPs are common at both location, in which days to 50% curd initiation recorded maximum number of SNPs when compared to the other trait. We detected 12 QTS, in which 5 for Delhi region and 7 for Barapani center on various chromosomes. This is the physical map and chromosome position of significant QTS which are associated with the curding trait Indian cauliflower. Coming to the salient findings of Objective 2, our study proved that there is an admixture in Indian cauliflower material from European cauliflower also, and 121 SNPs were identified, in which 38 from Delhi and 83 from Barapani condition, and 12 QTLs are identified, in which 5 for Delhi condition and 7 for Barapani region. Path ahead. These promising genotypes can be utilized for the breeding purpose for developing a varieties or hybrids having a good curding traits with respect to temperature, and the significant SNPs can be converted into robust marker for use in the marker-assisted breeding for better curding quality. And happy to say that this is the first detailed study on Indian cauliflower, and the identified QTL regions will be helpful for the breeders to map the genetic variation in curding trait to temperature response in Indian cauliflower. I have participated in many national and international conferences. Regarding publication, we communicated 2 papers to 2 different international journals like Science Horticulture and PLOS ONE, having a NAS rating of 7.96 and 8.78 respectively. I duly acknowledge to my chairperson, members, advisory committee, head, professor, faculty, division of vegetable science, IRN New Delhi, and genetic division, IRN New Delhi, director, ICR Research Complex for Northeastern Region, Meghalaya, Nahib Caste Project, PG School, ICR IRI, and ICR for JRF, and thank you one and all.
Thank you, Rakshita. Nice presentation. And this is open for discussion now.
Rakshita, nice presentation.
Thank you, sir.
The short maturing genotypes are susceptible to this stress. So how to address them, this challenge?
Sir, it means that in case of cauliflower, it is basically thermosensitive crop, sir, and it has 4 divisions like early, mid-early, mid-late, and late. Early groups have attended maximum. Early groups have attended early curd initiations when compared to the late groups, sir.
And what about the color of the curd also is important?
Sir. Sir, the early groups have orange, like basically cauliflower should be a white color, pure white color, sir. But in case of early cauliflower, it is like a somewhat orangish type, sir. Means a somewhat yellow color type. But in case of snowball and the mid-late group, it is purely white color. Sir.
Okay, thank you.
Yes, sir.
Is there any color change when there is a heat stress in cauliflower?
Sir. Sir. Actually, in case of mid-late, sir, some genotypes have showed it should be a white color, sir, and some genotypes have showed a little orange, like yellow color. Sir.
So it is purely because of heat stress.
Yes, sir. Because of higher temperature, because we have shown at the same time, sir, all the genotypes.
Can you just tell me how much of Indian cauliflower, in terms of other cauliflower, in terms of coverage in the country? How much of cauliflower cultivated is Indian cauliflower, and how much is other cauliflower?
Sir, in India, it is, sir, in case of North Indian condition, they will grow in like snowball groups, and in case of South India and
overall, in terms of percentage, can you tell me how much is Indian cauliflower which is cultivated and compared to others?
Sir, approximately 70% is Indian cauliflower and 30% is snowball cauliflower. Sir.
I was just now surprised. How did you link your first objective of heat stress with the second objective of simply finding out the relationship with coding traits? The genetic basis of coding traits you have done in your second objective. But in the first objective, or the major aspect of the study is heat stress related study, is it not? How did you couple both of these and come out with some kind of coordinated output or result?
Okay, sir. Sir, our major motto is that, sir, we need to develop a varieties which is having a plasticity to the temperature, sir. So that for our first objective, so we have selected 2 locations in which it has 2 distinct environment conditions, sir. So that we detected that how the temperature has effect on the various curding traits at Delhi and Barapani condition. And coming to the second objective, sir, we detected QTLs, so that, and we detected SNPs also. So by using that SNP, we can develop a varieties which is having
some kind of,
very good plasticity to the temperature
and some kind of presentation. Okay. Okay. All right.
Yep. You had detected some number of SNPs from Barapani and some from Delhi, right? These are the SNPs that are associated with curd characteristics?
Sir, 121 SNPs we have detected, sir, out of which 38 for Delhi region and 83 is for the Barapani region, which are associated for various curding traits such as days to 50% curd initiation and curd harvest and marketable curd weight, etc. Sir, these are all related to the curding traits, sir.
Why this difference between the 2 locations? It should be comparable, no? Or it can be different?
Said that we need to study.
Why this difference between the number of SNPs associated with curd characteristics at Barapani and Delhi?
Sir, I think different genomic region have contributing.
The
question is why you are getting different numbers of marker trait associations in Barapani and in Delhi. How many of them were common locations? How many were different, and why the variation is happening between 2 locations?
So 25 SNPs are common from both the locations.
Your genotypes are same, or the panel sequencing approach that you have used is same, that data is common. So what is different between Barapani and Delhi?
Delhi, yeah.
Sir, actually for Barapani condition, we have used only 91 genotypes, sir. For Delhi condition, we use 96
What is
genotypes.
different between Barapani and Delhi?
Sir, Barapani, the temperature is mild, sir. Like from
and you
June
are
to
studying
March.
the effect of temperature, right?
Yes, sir. Yes, sir.
So that is different, no? So if your genes are interacting with environment or not?
In environment interaction is there.
They are interacting, no? So if they are interacting and if environment is different, then this will change, will it not?
The another question, GBS predicts SNPs that are putative. They are not confirmed SNPs. Did you confirm them or validate them?
As no, sir, validation I will take next.
The
Because of
validation
time,
has to be done, no?
sir, but because of time limits, we will done PhD, sir.
I understand that. I understand that. Nice work. If there are any other questions.
How many market rate association you identified which were significant at say Delhi
12,
location?
12, sir.
12. And what is the maximum phenotypic variance explained by any one of this?
Sir, from Barapani Center, we identified 7, sir.
What is the maximum phenotypic variance explained by any one of
Sir, 31%.
31.
Yes, sir.
Okay. Now, if you wanted to identify this particular the gene in that region where you are getting marker rate association,
yes,
what will you do? That is responsible for this response. The temperature
sir,
tolerance.
again,
If you want to identify that gene, what will you approach
screening
now?
of markers again.
Screening of marker where?
So marker screening has to be taken, sir, to identify that.
Okay. Thank you.
Thank you, sir. And Chairman, sir, we move on to last presentation now.
Please.
And I invite Mr. Ankit from the discipline of Water Science and Technology.
Good morning, everybody. This is Ankit Deya from the discipline of Water Science and Technology. Now I'm going to start my presentation. As the Indian population increases, the projected water demand also increases. So with rapid pace of modernization and urbanization, the water will be relocated from agriculture to urban sector. So by 2050, increased demand of agri-product, but the lesser availability of the fresh water for irrigation. And at the same time, there is an increase in the generation of wastewater in both the urban and rural areas. And 62,000 million liters of the wastewater generated per day, but the problem is that only 37.5% of this is treated and rest is untreated. So what can be the solution for this problem? The solution is the use of the wastewater for the non-edible crop, and for this purpose, chrysanthemum, being the non-edible crop, is used for the research work. And it has other benefits like treating chest pain, reducing fever, treat allergies, and others, as you can see. Now there are some research gap with inadequate study on the following with the wastewater irrigation. So less investigation for the FDR sensor-based irrigation scheduling in commercial flower production, less studies on the impact of wastewater irrigation on the yield and quality, and at the same time impact on the nutrient and heavy metal accumulation. The title chosen for my research work is the standardization of sensor-based wastewater irrigation scheduling for the chrysanthemum. And with the following objective. First is this, to study the impact of the wastewater irrigation on the marketable yield and quality grading standard. Second is to assess the effect of wastewater irrigation on the soil health, crop water requirement, water use efficiency, and economics. And third is to identify the best value of the FDR sensor-based MAD of soil moisture for irrigation scheduling. In methodologies, we used experimental design as RBD with 4 application and 6 treatments, and number of the plots are 24 and variety is Thai-Chan-Queen. In treatment, we use 6 treatment, 3 with groundwater and 3 with wastewater, each with 3 level of the moisture depletion, that is 25%, 50% and 75%. These are different treatment plots showing the chrysanthemum. And we have used the FDR, that is frequency driven reflectometry, for this study, and it is the sensor that is used for the measuring soil moisture content at different depth. Soil sampling is done to analyze the effect of groundwater and wastewater irrigation on the soil physical, chemical and biological parameters. This is the pictorial view of different activities conducted during the experiment. Now results. Now first is the impact on the physical properties of soil. In physical properties, bulk density, it is found to be maximum in treatment 2, and the water retention at field capacity and water retention at permanent wetting found to be maximum in treatment 6. Now impact on the soil pH, EC and organic carbon. In all three cases, it is found to be maximum in treatment 5 with wastewater irrigation schedule at 50% MAD. Now impact on the available nitrogen, phosphorus and potassium of the soil, and it is found to be again maximum in the treatment 5, that is wastewater with 50% MAD. Now impact on the available micronutrients, that is copper, iron, manganese, zinc, and they again to be found maximum in treatment 5 with wastewater irrigation schedule at 50% MAD. And if we talk about the available heavy metal content in the soil, then they are found maximum in treatment 4, that is wastewater irrigation with 25% MAD. Now impact on the coliform bacterial population in the soil, and they have to be found maximum in the treatment 6 with the wastewater irrigation at 75% MAD. Now impact on the vegetative growth parameter of the chrysanthemum, and in this plant height and the flower stem length at harvest is found to be maximum in treatment 5, and the best grade stem deviation with the value of 2.5 cm is found in treatment 5 according to the Floral Marketing Association and Society of American Florist. Now impact on the stem strength, again according to the Floral Marketing Association Society of American Florist, if stem strength is close to 20 degree, then it is found to be better and it is found in treatment 5, that is wastewater irrigation with 50% MAD. Now impact on the flower parameter of the chrysanthemum, the flower diameter is found to be maximum in treatment 5, and the flower turgidity, that is relative water content percentage, is found to be maximum in treatment 4, that is wastewater application with 25% MAD, and number of the flower per plot is found to be maximum in treatment 5. Now impact on the waste life, the waste life is found to be maximum in treatment 5. In visual insect pest incidents, there are two treatment 3 and treatment 6, where termite attack is there because irrigation interval is more. There is no visual disease incidence, and in visual physical damage, some losing take place after irrigation, but the results are not significant. Now the impact on the flower stem yield, water efficiency and benefit cost ratio. All is found to be maximum in treatment 5, that is wastewater irrigation at 50% MAD. Now impact of NPK content in plant part, and NPK content in the leaf is found to be maximum in treatment 5, and NPK content in the flower is found to be maximum in treatment 6. Now this is the table for the computation of depth of irrigation for each treatment. In this we have calculated the three different MAD values, that is 25%, 50% and 75% MAD values, and accordingly for each plot we have calculated the amount of water that is to be applied. Now the impact on the production economics of the chrysanthemum, in this benefit cost ratio is calculated by the total revenue that generated from the spike production and the total variable cost incurred during the operation, and it was found that the benefit cost ratio is found to be maximum in treatment 5. Now the rating table. In this, the different parameter we have rated based on their positive and negative impacts of wastewater irrigation and groundwater irrigation on them. So we have rated the soil parameters. With the same rating we apply for the plant parameter to find out which treatment is showing the best results. So the rating score for the different treatment of chrysanthemum experiment, and it was found that by computing both the soil parameter and plant parameter, so treatment 5 is found to be best in all the treatments. So at last I conclude to say that the quality of both groundwater and wastewater is safe for irrigation, and wastewater irrigation application in chrysanthemum does not attract any insect pest and disease incidents. Wastewater irrigation scheduled at 50% might have significantly enhanced the flower size, yield, water efficiency and net returns in terms of benefit cost ratio, And the standard value of MAD for irrigation scheduling chrysanthemum found to be 50%. That result in the higher quality and productivity of chrysanthemum without degradation of the soil fertility. So future recommendation, the experiment on FDR sensor based irrigation scheduling can be standardized at more precise value of MAD like 10%, 20%, 30%, etc. FDR based scheduling can be standardized for other flower crops and varieties using this MAD approach, and different type of soil moisture sensors can be standardized for irrigation scheduling flowering crops, and in future irrigation scheduling experiment can also be undertaken with drip irrigation system. At last, thanks to my chairperson Dr. Dhara Singh Gujar and members of my advisory committee and also project director and Professor Dr. Man Singh sir and other faculty members, Water Technology Center and also Division of Agronomy, Dr. S.K. Jha sir, Professor, Division of PhD, PG School, IRI, my parents, friends and Gokul, my classmate who helped me. And last I am closing here, and thanks to everyone again for listening, and thank you. Jai Hind, Jai Jawan, Jai Kisan. Thank you.
Thank you, Ankit.
Thank you, sir.
Open for discussions.
What do you understand by wastewater here, Mr. Ankit?
Sir, we are using here the municipal wastewater.
Municipal wastewater.
Yes, sir. That contains
municipal wastewater. What are the characteristics of municipal wastewater?
Sir, that does not have the more number of the heavy metal that can be applied for the crops. Like we are using chrysanthemum, that is the non-edible crop, so we can use this wastewater because it is only for the ornamental purpose, ornamental crop. So we are not eating that crop.
Not like that. Continued use of wastewater containing either acidic or high soluble salts might in the long run cause salinity or soil related problem. Have you just looked at that? Because it's not just one season like that. Continued use of these wastewaters probably in the long run might lead to problems, soil related problems. Could you throw some light on that?
Sir, we are using this wastewater in our experiment, but we have seen that continuous use of wastewater lead to heavy metal accumulation in plants also, soil also, and so continuous use of wastewater in the same field cannot be sustainable for the soil also and plant also, sir.
Thank you.
Thank you.
Sir, can I ask a question?
Yes, sir.
Mr. Ankit, you have measured relative water content in different water treatment levels.
Yes, sir.
So, and you found that wastewater treatment, the relative water content was higher, and in other water treatment, relative water content was low.
Yes, sir.
So, how will you explain that? Because if there is a similar amount of water availability, you expect that relative water content should be the same.
Sir, it may be due to the favorable effect of organic matter that at 25% depletion, when we regularly apply water, so the treatment with 25% MAD value can absorb the water. And
when you calculate on percentage basis, if it contains more organic matter, then water available should be less when compared to the soil with same soil with less organic matter, because when you add organic matter and other thing, it will interact with water. So the matric potential, it will increase the matric potential. So rather at same soil moisture, available water should be more in the other kind of soil, or any other reason you think that it is there?
Sir, in wastewater application, the favorable conditions created by the wastewater, because nutrients are also there, and we are applying water at 25%. So regular apply of water, then it is more available to the plant. So that this may be the reason, sir.
Okay, thank you.
Thank you, sir.
What is MAD, Ankit?
Sir, it is a manageable allowed deficit or maximum allowed deficit. Sir, we have to understand two things while understanding the MAD. Field capacity 100% available water is there, and permanent wilting point 0% available water is to the plant. But when water is reduced from the field capacity to permanent wilting point, there is some point that yield start reducing.
So the point where the yield start reducing, we can't wait for the permanent wilting point because their yield, where plant will die. So at in between the stage, there is some stage that yield start reducing, and there is the state we have to apply water. Sir, this is the mad value. So
how
that
do you
the
determine that?
Sir, we use FDR moisture sensor for this, sir.
Okay.
At different depths, sir. Yes. Yes, sir.
You used the phrase flowering crops. You meant flower crops or flowering crops?
Sir, these
in the conclusion part, you said flowering crops.
Sir, these ornamental flower crops, sir.
Flower crops, yeah. So,
yes,
yeah.
sir.
Okay. What is the practical outcome of your work?
Sir, practical outcome is that as we are living here in close to NCR region, and here plenty of wastewater is generated. This is a tier 1 city and tier 2 city. Main problem is this wastewater. So we can use this wastewater. And sir, regarding farmers are, I'm not saying that farmer will buy the FDR sensor that is lakhs of rupees. So we have seen practically, I have seen practically that 25%, 50%, and 75% depletion. So we should irrigate at 50%. So how we can know that 50% irrigation is it? We say that 25% depletion reached at 3 to 4 days after irrigation, 50% irrigation reached at 7 to 8 or 10 days after irrigation, and 75% depletion reached at 40 days. So we will advise farmer that you should irrigate around 7 to 8 days irrigation interval, so that it will result in the better yield and better flower productivity. And at the same time, if you have option of the wastewater close to NCR region, then it will provide a good market for these flowering crop. And it is we can also use this with wastewater, because sir, here wastewater is plenty, sir.
Again, Ankit, you said flowering crops.
Oh.
You got some hangover on it, huh? Yeah. Any other questions from the panel of judges or from the audience? It seems audience says, still 347 people are connected. Audience has been quite silent, it seems. But anyway, that would have been. Any other questions? No, then Professor Jha, we should thank Ankit, and we could wind up for the day.
Thank you.
Yes, thank you, sir. Thank you, Ankit.
Thank you, sir.
We have come to the concluding part of the day. We have finished day 1. The 2 days we have for merit medal presentation. So we have concluded with 21 presentations today of MSc and MTech students. Before we proceed further, any remarks from Chairman sir, or
for these people who presented?
Yes.
Or
yes,
for the people who presented?
Well,
Yeah,
sir.
So you have them, or you would like to? They can be there, the students. Or you want them to log out?
No, sir. It is just a general.
just a general general
All right.
observation and general.
Fine.
my general impression about all the presentations taken together was this, that the young crop of students that is coming out is really very keen and committed to the work they are doing. They believe in what they are doing, and they also seem to know the way they can get around doing what they desire to do. The quality of slides was very good. The work they have done is excellent. And the way they have presented each one of them sounded like a professional narrator. So they have really worked hard on that. And I was highly impressed by the way they have gone about it. There are a couple of things that I would like to point out. 3 points generally that applies to most of the presentations. One, that each one of them has started their presentation with a prolonged introduction that sounded like a, let's say, popular presentation initially, and then it narrowed down to a scientific presentation. To me, when I started listening to the initial part of the talk, I thought they were going to talk about something different. And it was a kind of general information presentation that I was getting into, not a highly specialized this. So this is a old-fashioned person's observation. May not tally with the viewpoint that the present generation holds. But I think for people like us who tend to drift out of the subject into some other thought, it's much better to get into the grill of the subject immediately rather than let them drift into what you start talking about COVID, you start talking about pandemic, you start talking about something else. And then people like me have a tendency to get the hint and get into some sideway and may miss the emphasis of your talk altogether. The other thing was that almost all the slides that were presented contained too much of information in a single slide. The students have done lots of work, lots of distillation of their work into a narrative, and I think when they present, their focus should be to convey to the judges not how they did it. They can hint how they did it. If judges are interested, they can ask about the method subsequently. But what really came out of it, and what was the interpretation that came out of it, where their intellectual input has gone into. So that will reduce the amount of information presented into a slide, and the importance of the finding would be more easily communicated if it was communicated more directly and with little distracting information in there. For this reason that they have devoted substantial part to introduction, they have too much to talk about. Some of the students really talked at a pace which was very difficult to keep pace with them. What they are saying, very rapidly they talked about it. Unless you are familiar with the subject, it's not easy to keep walking with them, or running with them rather in this case. So this was my general observation, but highly satisfying and highly revealing discussion and presentation. So I was very happy. I am very happy that I was a part of it, part of the audience listening to them. But the way they presented, I will say again, it sounded like a professional narrator. They could be a narrator anywhere today. My judge colleagues may have observations to share.
Yeah, at the outset, as indicated by Honorable Chairman of today's panel, I think too much of diffused information was put into one slide. Rather, it could have been better had it been in the form of bullets, which convey meaning directly. As indicated by Chairman, that part of procedural these things that could have been kept in the background for the audience and panelists to ask the questions. And introduction was, I thought, a bit lengthy, could have been reduced. More emphasis could have been on the results, but at the same time, just they are entering into the career, we should we appreciate their quality work. And I think almost all of them have published their articles in NAS rating more than 6 or 7. That itself speaks of their determination and dedication towards work. Probably since they are in the cell division metaphase stage, as the time proceeds, probably they'll become good experts. And I'm sure with the increase in time and with more exposure to such scientific meetings, probably they'll refine themselves. And the quality of work was very good. And this has laid a sound foundation for them for a bright future. And I'm sure, looking at the work turned out by IRA students, the nation will be in safe hands. Thank you.
Thank you, sir.
Actually, I found it very difficult to differentiate. It's embarrassment of riches, and that's an honest opinion.
Yes, sir. All superlatives, sir. Everybody, competing with each other in quality work and publications, and at master's level, attending so many conferences and getting so many prizes. That itself speaks of their dedication and determination, sir.
So if someone gets a few marks less or a few marks more,
yes, sir.
It's only
it's all superlatives, sir.
As many would say, the ignorance of the judges that crept into.
Yes.
I think majority of the presentations are outstanding, and the students are very confident. It also reflected how the guides are also planning their work. That also it has reflected very clearly with the new kind of problems they have allotted to the students. And as it was pointed out by Honorable Chairman, last time also it was told that introduction part, they should focus straight away on their problem rather than giving more introduction. That is one thing. And most of the work, it showed their commitment, and there is almost, it's very difficult to judge and a very difficult task. They have done outstanding job, and I am very glad to be part of this panel. Thank you very much.
Thank you, sir.
Sir, as usual, I think the students have been wonderful. I mean, they have put in a lot of effort as usual. IRI students, most of us have been associated with IRI. So we know they put in a lot of effort, and the chairpersons and members of the advisory committee also put in a lot of effort, and it's evident from the presentations that we have seen today. I do agree with the chairman that and the other members that the information is, some of the slides, the information was too much. So, but one thing that I would agree with the chairman is that some students were very, very fast. So as he said, sometimes for people from other disciplines like me, it becomes difficult to follow what's going on. So that's a problem. But in general, I would say the students have been very nice, very well presented their work. So good luck to them.
Thank you, sir.
It is actually same as how we do in the normal every year convocation. So I don't find that there is any way it is different from what has been going on in every year. But one thing I can say that every year things are improving, especially in terms of the publications by the students. Otherwise, earlier we never used to find publication from MSc students on a very highly rated journal, not even peer-reviewed journals. So one way things are improving, and but of a late it is becoming almost stereotyped. That is why I think, as chairman has also felt, there's a need for some kind of variation among the presentations, and that will come only when you avoid this kind of introduction, as has been pointed out by him, and then come straight away, maybe use 1 or 2 slides in the initially, and then come straight to the crux of the problem, and then tell only significant finding. That should have been much more better than especially what they already published. They should have focused on that. Otherwise, overall, I feel that the contributions by the students and the faculties associated with them is every year, at least in that particular case on entomology, I could see a good transformation in the way things have been extrapolated in the pure aspect of taxonomy. So I feel that things are improving. Only thing is they have to be modulated now, and then seen that they are regulated towards an improved way of presenting, so that we can improve this, we can remove the stereotyped way of viewing it, and there is some variability. So that will be good. Otherwise, all are very good. In fact, I have learned a lot by attending this one, and I wish all of them good luck.
Thank you so much, sir. So we are at the conclusion of the day one. We have heard the chairman and the members of judging committee. We have noted down the students and faculty members also, the suggestions given. Sir, this has been, as I had started in the morning, a rich tradition of IERI. This day is full of energy. The BP Pal auditorium is sounding with claps after every presentation. That was missing because of this pandemic. We are through virtual mode, but I hope all of you will agree this was a very energetic and powerful presentation throughout the day. All the 21 presentations were excellent, and we appreciate the efforts that take to make such presentation. So congratulations to all the students for making this day as it used to be a regular practice in BP Pal auditorium over the years. I would like to thank now, it is my duty as a convener of this session.
Dr. Jack, before that,
we can clap for the students now. Please, we can do that.
Brief remark from the dean as well as a few words from director.
Sorry.
Yes. Thank you, Dr. Jha and Dr. Manjaya. And really, it was a great day today. As Dr. Jha also mentioned, that we were just missing in the auditorium the presence of all the students and the claps, which we used to hear every year. And as I said in the beginning itself, COVID-19 pandemic has brought a very great change, a transformational change in all of us. And this is also one of the changes that we have accepted it, and now we are doing it through virtual mode. And the comments or the observations given by our judging committee members and our chairperson judging committee are really very, very relevant. And I hope all the faculty members and the students, they will be considering them in future. And during the presentations also, we could see that all the members, they were very thoroughly understanding the presentation and asking very relevant questions. So that was really talks the wisdom of all the judging committee members. So the students also should take home the message that in future they have to be very crisp, and briefly they should introduce a research topic and come to the main significant achievements straight away. So, sir, we profusely thank you, sir, all the jury members for accepting our invitation, and throughout the day, taking from morning since 9:30, we are all here, and all of you are throughout listening to all the presentations and giving your expert opinions and comments. So we are profusely thanking all of you and all the faculty members and all the others who joined since morning. So this is another day is there tomorrow, and we will continue the same proceedings for the PhD merit medal student presentation tomorrow, sir. So with these words, I am again thankful to all of you and wishing you good day ahead.
Thank you, ma'am. May I now request our director, sir?
Thank you, Dr. Jha. And it has been a great pleasure listening to all the presentations and the critique and the compliments that have come from the honorable members of the judging committee and the chairman. Both have to be taken with equal seriousness. And there's a lot to improve, particularly what Professor Murthy said and what Professor Vidi Singh said. There is a lot of value in that. The kind of accolades that came from the members and from the chairperson is something really great. At least I can say for myself that I didn't get this kind of compliment when I was in MSc student with Professor V.D. Singh. So I must say that these students have really done extremely well and evolved. And that's a great satisfaction for all the teachers who have worked for the students and the students themselves, working so hard and then, putting the information in a way that is comprehensive, presentable, and appealing to everyone. And I hope tomorrow in PhD program, there is some lead from today's comments. And if there are some students who have joined, if not, at least professors can pass on the message. I can feel that there is no time for making a change in the presentation, but at least while making presentation, they can articulate themselves so that some improvement in the introductory part can be done. But you will find a clear difference between the MSc and PhD presentation as far as that part is concerned. MSc is more enthusiastic and they have more bubbling ideas of the kind of presentation that you have seen today. So I hope that tomorrow we are going to have another great day. And although Professor V.D. Singh is not in that panel, I would request him, if he has time, to also see the presentation by the professors of these students who have presented today. And professors are going to present the students of all other, the work of all other students, because we are having only one from each division. So the professors will be making presentation of the students who could not make it to this place. Their work they will be presenting, because all work is significant. So that would be again a good exercise that will go for 2 days after tomorrow.
9th and 10th. Right?
9th and 10th.
I'll join somewhere. So, Daksa, before you close, just one thing. There is a story, Ashok, I'll share with all the members. This relates to Vasisht and Vishwamitra. So one Purnima night, the full moon was there, bright moonlight. And Vasisht was having a discussion with his wife about Vishwamitra. And Vasisht said, wife said that, look, Vishwamitra is shining as the moon is shining. So Vasisht said that, look, you have made a mistake in this comparison. The moon has a shadow. Vishwamitra doesn't have that shadow. And at that point of time, Vishwamitra was waiting for the time when Vasisht will sleep so that he could assassinate, because he was not getting the praise from acceptance from Vasisht. And then Vishwamitra realized that this is what is expected of him. It's not the moon, the sun. So maybe the lack of appreciation for you, Ashok, is because of you have to still shine brighter. If there is any space for that brighter.
Thank you, sir. Thank you so much.
Just a suggestion, Dr. Rashmi, for tomorrow's and the lack of claps that you were mentioning.
Yes.
We can have a recording of claps also and play it at the background. That will complete the whole scenario.
Yeah, let's see.
We can.
We can.
We can have a clap now.
Clap.
So can
Now
we?
at least our physical clap can be there. We have.
We have to unmute ourselves, each one of them. We can have.
Yes, yes. So it can be a nice clap for all the presentations which students gave. Excellent.
You can hear that. Good.
Thank
So
you,
that
madam.
clap has to be built in each presentation, after each presentation. It used to happen.
We can.
No?
We can.
We can do that.
We
but
can
I hopefully we don't we would not need it for next year, but for tomorrow if possible. Next year we will be together physically.
Yes, sir.
Right. Thank you so much, Dr. Jha. Over to you, unless we close for today and join tomorrow at 9:30, right?
Yes. Thank you, sir.
Thank you.
So lastly, I present my vote of thanks. I first of all,
वो तो अभी कल ही कर दीजिएगा ना?
कल ही कर दूंगा, ठीक है।
और क्या? अभी तो
कल फाइनल करिए।
फाइनल कल है।
So thank you all for joining today.
Thank you.
And we meet tomorrow again at 9:30.